Perfect, thank you. I wanted to start by introducing myself. So my name is Ben Moore, and I am the Ensemble Outreach Project Manager. So that means that the team that I work in is responsible for delivering courses and training for people who want to learn how to use the Ensemble Genome browser. We also answer a help desk, so we provide online support for people who have questions about using Ensemble, and we help people use Ensemble that way. And we also are in charge of the social media channels and blog announcements about the Ensemble Genome browser. So many of you I may have seen before. We had a workshop associated with Noblekinmat earlier this year in February. So the idea of this workshop is to build on the workshop that we covered in February earlier this year, and to extend a little bit further and thinking about some of the other data types and some of the other ways that you can access the data and use Ensemble. So we want to start off today by reviewing what we covered in the last workshop. So I'm going to introduce you to the Ensemble project and some of the main data types that we have in the browser, including genes and transcripts. And I'm going to remind you how you can navigate around the Ensemble browser to access that data. So if you didn't attend the workshop in February, then that's not a problem at all. Hopefully that first session will serve as a good introduction for you to get up to speed with everyone else as well. Then what we're going to do is we're going to move into exploring gene regulation data in Ensemble. So we're going to be thinking about how we can use Ensemble to explore information about features that regulate gene expression. Then we're going to have a break in the mid-morning. And then after the break, we're going to move on to our final two modules. So we're going to look at how we can visualize our own data using the Ensemble Genome browser. And then finally, we're going to move on to a module that we call Advanced Access. So this is thinking about some of the ways that we can access Ensemble data not through the web browser. So all of the modules today are going to be thinking about how we can use the online genome browser through our computers. But there are some other ways that you can access larger data sets, not through the online web browser. And then at the end, we're going to have time for some feedback, and then also time to answer any questions that you might have as well at the end of the session. So each of these modules has a particular structure. So we normally start off with a presentation that explains the background about where the data has come from or how the data has been produced in Ensemble. Then we're going to move on and have a demo. So I'm going to walk you through the different pages of the Ensemble Genome browser, exploring the data and seeing how we can manipulate the views to see the data that we're interested in looking at. And then finally, we'll have an exercise session. So there's exercises for each module, and they just give you a chance to try out the exercises for yourself. So the exercises are not a test. They're not going to be graded or marked. It's just a way for you to begin exploring Ensemble for yourself, clicking on the different links on the pages and just getting familiar with the way that the pages are laid out and where the data is. So there's lots of exercises for each module. So I'm not expecting you to complete all of the exercises for each module. The best thing to do is to look through them at the beginning of the exercise session. Look at the ones that are the most interesting to you, because they all focus on different species and slightly different data types. So the best thing to do is to look at those exercises, pick the one or two that are the most interesting to you in your areas of research and have a go at those. And then if you have time, you can move on to the others as well. So the first thing that I wanted to make sure that everyone has before we start are the course materials. So I believe that Ashraf has emailed the link around to you all with the training materials, but I just want to... sorry, I'm just getting some messages saying that the audio is not clear. So I am...
it's clear from my end. I
Okay.
can hear you clearly.
Okay, so I will try and speak up and speak maybe a bit more slowly. Maybe it's a problem with the audio at a few different people's ends if other people can hear me. Okay. So Good. Okay, so I think it's just a couple of people. So maybe it's maybe your internet connections, or maybe you need to leave and join again, if that might help perhaps. Good. So yeah, as I was saying, the first thing that I want to do is make sure that everyone has the course materials. So we've put all of the training materials for this workshop online. So there's a website that you can see at the top of my slide here, training.ensemble.org. So this is our Ensembl training pages. I'm just going to jump out of the presentation and show you how to access the materials. So if you open up a tab in your internet browser and type training.ensemble.org into the address bar, I'll copy the address and put it into the chat box for you all to find. So you can click on this link in the chat box, and this will take you to the Ensembl training pages. So this is not the Ensembl Genome browser. This is the pages that we have that contain all of the information about the workshops and the training events that we're running. So if you click on from the home page, if you click on the link in the bottom left-hand corner with the magnifying glass here, this will take you to a list of all of our training events. And you can see at the very top, we have this entry in our calendar for the Ensembl browser workshop, Noblekinmat. So this is our event for today. So if you click on this link, this will take you to a page that has all of the training materials for our course today. So you can see some information about the course. Over on the right-hand side, we have the links for some of the materials. So you can see the first link is for the presentation. So this is a PDF of the presentation slides that I'm delivering. So you don't need this during the workshop today, but you can download this and save it for yourself to look at later as a reference. Underneath the presentation, there's a link for what we call the living document. So if you click on this now, this takes you to a Google document that has information about the course and is a really good platform for also asking questions as well. So if you scroll down, you can begin to have a look a little bit at the learning outcomes for our workshop today. On the second page, you can see a rough schedule of the morning that we have together. You can see more information about the trainers. We have webinar slides. So this is the link for all of the training materials. The feedback survey, there's a link that I'm going to tell you about at the very end of the course today. And then also, I'm going to add a URL of the workshop recording. After the workshop and I've processed the recording, this will be added here as well. There's just some rules about online training, so how we can all behave in the Zoom call to make sure that everyone gets a chance to ask questions and to make sure that no one is interrupted. And then finally, there's some links for the different resources that we're going to be using throughout the workshop today, and some further ones as well. At the very bottom of this document, you can see this section called questions. So this is a really good place for you to ask questions that you have about the course today and about the materials that we're covering. So if you have a question, the idea is that this is a document that everyone has access to edit and to view. So if you have a question here, for example, you can just enter your name and then you can write your question here. So you can write your question, and then you could even perhaps include your question here. You could have a screenshot, it may be if you need to show me something, or a link to a page that you're having trouble with. And then what that will mean is that myself or Alina, who's my colleague, who I'm going to introduce you to in a moment, can add our answers in here. So we can give you the answers to your question, and even if there's follow-up questions, then we can continue the conversation in this document as well. The idea is that these questions, therefore, everyone can see, and everyone can see the answers to as well. So what this means is that this document sort of evolves over time, and it becomes a knowledge base where you can always come back here and see the answers to the questions that you had during the session and that other people had as well. And you can use this into the future as well. So I'm just going to get rid of this example that I wrote here. So yeah, please do I really encourage you to use this document and to add your questions in here. Obviously, you can use the Zoom chat box if you want to, but the problem with that is that obviously if there are lots of questions, it can be very difficult for me and Alina to track the different questions and to see if everything has been answered. So this is a much more organized way to have your questions asked and answered. So I can see there's lots of people in this document already, so I'm glad that people are able to find that okay. The final thing that I wanted to show you with regards to the training materials for today is this section at the bottom called Demos and Exercises. So this is where you're going to find information about our walkthroughs and the exercises that I'm going to ask you to do as well. So these are the three modules that we're going to be covering today. The first one is called Overview. That's the recap, the review that we're going to do. And you can see that if you click on the Overview title, you then open up this two panels called Demo and Exercises. So if you expand the Demo panel, you can see that we just have a walkthrough, a description of the walkthrough that we're going to have today. So this is not, you can obviously follow along with me live when I'm doing the demonstration, but this is here to act as a reference that you can look back over as well. If you open the exercises panel, you'll be able to see that we have the exercises that I'm going to ask you to do at the end of each module. And then obviously, as well as the exercises, you can also see that we have some model answers and also a video answer as well. So if you're not sure about the answer to the question, or if you get stuck, you can always come here and see the answers and see how we got the answers as well. The final thing that I wanted to mention is that these materials are going to be available throughout the workshop today, but also into the future as well. So if you save the link for this page, you can always come back here and find these walkthroughs, find these materials, and use them as a reference in the future as well. Good. So hopefully everyone has those training materials. The final thing that I'll do, I'll just quickly pop this link in the chat box. This is the link for the actual page of our training event. So hopefully you will have that. Sorry, is there a question? I think someone might have just unmuted themselves there, but yeah. Good, so that's the training materials. So just thinking about today, I want to reintroduce you to what Ensemble is, thinking about the different types of data that we can get through Ensemble, and then learning how we can navigate around the Ensemble browser to access that data. And then finally, I want to also show you where you can go to get further help and documentation as well. So they're the learning objectives for today. So I've already spoken about this a little bit, but if you need help or if you have any questions, there's a number of ways that you can get help. So the first thing that I need to do is introduce you to my colleague, Alina. So Alina's joining us as well today. And obviously I'm going to be doing some presenting and walking you through the main parts of the workshop. But Alina's going to be on hand to help to answer any questions, just in case there's so many that it's difficult to present and answer the questions at the same time. So Alina will also be answering some questions in the chat boxes. So please do ask your questions in the living document or in the Zoom chat as well if you find that easier. If you have questions after the workshop, then you can email our help desk @ensemble.org. This is where you can ask questions that we'll work to find out the answers for you after the workshop. So if you do, well, what I'd like to do now is just quickly ask around to see if there are any questions about the format and the layout of the workshop and the materials before we start the workshop itself. Okay, so everyone, there's no questions, it doesn't seem, so everyone seems very happy. So let's get on with the workshop. So I want to start by introducing you to the Ensemble project and some of the background. So the Ensemble project started just at the turn of the century, so in the late 1990s and the early 2000s, just when the first draft of the human genome was being sequenced and published. So that was when the Ensemble project started. Obviously, DNA sequencing was actually developed and was occurring around the world before then. So the first organism to have its genome sequenced was a bacteriophage. Okay, so again, I'm getting some more feedback saying that my voice is breaking a bit and it's not very clear on the audio. So hopefully, I think this might be just a problem with a few people in the workshop today, and maybe it's the microphones and the speakers at their end, because I think the majority of people can hear me okay.
Yeah, I can hear you.
Okay, perfect. Okay, so the first organism to have its genome sequenced was a bacteriophage back in 1977. Fred Sanger and his team here in Cambridge in the UK, they developed a way to sequence the genomes of simple organisms. So they sequenced a bacteriophage which had a genome of just 5 kilobases in size. So by today's standards, that genome is obviously very, very small, but that technology laid the foundations for more and more complex organisms to have their genomes sequenced until in the early 2000s, in 2004, we had the publication of the first draft of the human genome sequence. So this was 3 gigabases in size, so 3 billion base pairs is the size of the human genome. But even since then, we have the advent of next generation sequencing technologies, which has meant that species and individuals can have their genomes and their exomes sequenced in a very, very, very rapidly and for a very much lower cost than the first human genome draft was that costed to produce. So what we have now is an explosion of data. So we have a number of different projects around the world that's aiming to sequence basically all of life on Earth. So there's a few projects, for example, just to mention. So there's the Darwin Tree of Life project, which is a project to sequence all of the eukaryotic species in the UK. And the idea is that all of these species will have their genome sequenced and annotated and then presented in Ensembl. Obviously, presenting with Noblekinmat today, I know that this is a worldwide initiative, but I think a lot of you guys are joining from Africa today. There's a recent project called the Africa Biogenome Project that's just starting out, that's aiming to generate sequences for lots of different indigenous African species and to get their genomes sequenced and annotated and available for everyone around the world to use. So at the moment, there's lots of worldwide initiatives and collaboration and work to generate the genome sequences of lots of different species on the planet. So this is really why we need genome browsers, because we have so much data that's being produced. But as scientists, we need a way for this data to be organized and annotated so that we can really make use of it and to understand it. So when a genome is sequenced, this is generally what comes out of the sequencer. So it's just a string of A's, T's, G's, and C's. And without any context or without any annotation, this sequence doesn't mean anything to anyone. So this is what Ensembl does. So we take this genome sequence and then we annotate it with a number of different types of features that are obviously interesting and useful for us as scientists. So we might annotate the genes, for example, which are made up of their different isoforms or transcripts. So we annotate the genes and transcripts, which obviously include exon and intron sequence, for example. We also regulate features that control the gene's expression. So we might annotate promoters or enhancer features that are able to regulate the expression of different genes. We also annotate variants that might be linked to phenotypes and diseases and those sorts of things. So this is exactly what I want to show you through the course today, is how Ensembl takes this sequence data and we annotate it to make it useful for everyone around the world to use in their work. So Ensembl has 4 main types of data. We covered some of them in quite a lot of detail in the workshop in February. We're going to revisit them all again today, and then we're going to move on to some others as well later on in the session. So the first type of data is the genomic assemblies. So it's very important to remember that Ensembl is not a sequencing project. So Ensembl doesn't do any of the sequencing ourselves. We rely on worldwide collaborations and research groups and initiatives to sequence the genomes of different species, and then to deposit those genome sequences into publicly available archives like GenBank or ENA, which I'm sure you might have heard of before. So we take those genome assemblies and we annotate them with an automated gene annotation pipeline. So that's the first type of data we have. It's the gene and transcript annotation. The second type of data is the variation data. So again, we draw data from a number of different variation databases that include both small and large scale sequence variation data, including things like allele frequencies and phenotypes as well. The third type of data is the comparative genomics. So these are analyses that we perform within Ensembl on the genome's sequences and the genes that we have annotated. So this is where we compare the genes and the genome sequences from one species to another to predict things like gene trees, homology relationships, and alignments and conserved regions. The fourth type of data is the regulation data. So this is where we annotate features that are able or predicted to regulate gene expression, such as promoters, enhancers, and repressors. At the moment, we only have this data available for human and mouse, but we're hoping, and I'm going to explain this annotation for human and mouse later on today, but we're hoping to extend this annotation to other types of species in the near future when this data becomes available as well, such as DNA methylation data. We need this to do the annotation. So to draw these four branches of data together, Ensembl has a number of features. So we have what we call the gene build or the gene annotation. This is actually old now, so it's actually over 300 different species that we have in Ensembl now. And then we also have obviously the variation, comparative genomics, and regulation data display as well. So these are our four main types of data for all the different species that we have. The other things that you can do is you can display your own data in Ensembl alongside the publicly available data in order to begin visualizing and analyzing your own data. And I'm going to show you how to do that later on this morning. We also have a number of tools for data processing and analysis, such as the variant effect predictor. We covered this tool in February, so I'm not going to look at that again today, but you can read more about that through the documentation and the recordings that we have. Then we have some tools for data export. So we have the Bioinformatics data export tool. We did cover this again in February as well, so I'm not going to look at that in detail today. But we also have access to the Ensembl data programmatically through the Perl and the REST API. So I'm going to touch on those at the very end of the workshop today for those people who are more able to use the command line and to write computer scripts to access the data. The best thing about all of this is that it's completely open source. So all of the data that we have in Ensembl is completely free for you to take and use in your work. So as I mentioned, Ensembl has access to over 200 different genomes with annotation in Ensembl. And this is just a species tree just to show you the range of species that we have in Ensembl. So if you were to, obviously, I don't know if you can even see these species very clearly from your screen, but the idea, the important thing to notice if you do look at all of these species is that Ensembl is dedicated to vertebrate or chordate genomes. So we only have species in Ensembl that are the vertebrate species, so those that have a vertebrae or a chordata. We don't have any non-vertebrates in Ensembl, apart from three. So we do have Saccharomyces cerevisiae, baker's yeast, Drosophila melanogaster, the fruit fly, and also C. elegans as well, the nematode worm. So these are three model organisms that are important in biological genetic research. So we do include those three species in Ensembl as well. But then we obviously have human, and then a large number of other great apes. We have all the other great apes, and then we also have the other primates and placental mammals, including rodents, and agriculturally important species, evolutionarily important species, and conservationally important species as well. And then we have also a large number of other birds, reptiles, fish, and amphibians as well. So as I mentioned, Ensembl is a project that's dedicated to the vertebrate genomes, and you can access all of that data through www.ensemble.org. So this is our web page where you can find all the data. But we also have a sister project called Ensemble Genomes. So this project was a bit younger than Ensembl. The Ensemble Genomes project started around 2007, 2008, and it's a related but separate project that's devoted to non-vertebrate genomes. So if you're studying bacteria or fungi or protists or non-vertebrate metazoans or plants, then you can access all of this data, but through the Ensemble Genomes pages. So it's slightly confusing because you have to go to two different pages depending on whether you're studying a vertebrate or whether you're studying a non-vertebrate. But once you get the hang of it, it's not, you just have to remember at the beginning. So if you're studying a vertebrate or like human or mouse or zebrafish, for example, you come to Ensembl. If you're studying a bacteria or a plant or a fungus, then you just come to ensemblgenomes.org. The pages are laid out in a very similar way, so you should be able to cross-reference, and everything that you learn today should be applicable whether you're looking at Ensembl or Ensemble Genomes as well. So before we start our first demo, I just wanted to introduce you to our gene and transcript annotation methodology that we have in Ensembl that I spoke about just earlier. So when you're looking at genes and transcripts in Ensembl, you're looking actually at the product of our gene annotation pipeline. So we have two different gene annotation methods, and they are used differently depending on the species that we're looking at. So if you're looking so every species in Ensembl has had its genes annotated using an automated gene annotation pipeline. And we're going to think about this pipeline in a bit more detail in a moment, but the idea is that this automated pipeline uses a range of scientific and biological evidence, aligning it to the reference genome and using it as evidence to predict the genes and transcripts. So these might be things like RNA-seq data sets or protein sequences. It aligns all of these sequences to the genome and uses that as evidence to predict the location and the model of the genes. For a small number of other species, we also have manual annotation. So for human, mouse, rat, and zebrafish, we have manual annotation of the genes as well. So there's a group called Havana. They're a group of maybe 10 or 12 curators that metaphorically walk along the genome, annotating the genes. Again, using a range of biological and scientific evidence that I'm going to speak about in a moment to annotate the genes as well. So the idea is that if you're looking at a species such as human in Ensembl, the genes that you're looking at are actually the product of these two independent annotation methods. So you have the automated annotation and the manual annotation, each annotating genes in their own way. And then we bring the output of those two pipelines together to produce our final human gene set, for example. For all of the other species that don't have the manual annotation, the genes that you see will just be the output of the automated annotation. There's a couple of questions that have come in on the chat box. So I'm just going to leave the presentation for a moment. I'm going to just try and answer these questions quickly before we move on. So the first question I can see is from Mark John, and it says, is Ensembl database different from NCBI? So the answer is yes. NCBI is a database that's hosted and maintained from the US, whereas Ensembl is based at the European Bioinformatics Institute here in the UK. The underlying data in the two different databases is similar and even sometimes exactly the same, but some of the ways that the data is processed and analyzed is different. So for example, if you're looking at the human genome sequence or the genome for a number of different species, then the genome sequence is exactly the same, and it's shared across these two databases. However, then there's the gene annotation, for example, is done independently by Ensembl and by NCBI. So sometimes when you're looking at the same gene in the two different databases, there might be some slight differences that you need to be aware of and take account of when you're doing your analysis and your interpretation of your data. So while these two databases are separate, Ensembl and the EBI does collaborate with NCBI very heavily. So what we always try and do is we try and provide cross-references to tell you that, you know, this gene in Ensembl is the equivalent of this gene in NCBI. So we give you the cross-references and the IDs for different databases, so you can always compare from one to the other. So hopefully that answers your question, Mark. And then there's a question from Saba. The question is, did genome a DNA? So I'm not too sure exactly what the question means, but I think what you're asking is when I say genome, do I mean DNA? And the answer is yes. So obviously the DNA sequence that is so within a cell in an organism, there's obviously the DNA, like the chromosomes, which make up the genome. So that all of the genetic material in the nucleus of the cell is what we call the genome, and then the DNA is the actual double-stranded polymer that obviously that sort of is wound around and builds up, that forms the genome. So the DNA is the actual helical structure, and the genome is the name for all of that genetic material, the DNA sequence put together in the nucleus. So that's what we have in Ensembl. We have the sequence of the DNA, which forms the entire genome. Good. So that's the two questions that I can see in the chat box. So if there are no other questions, then I'll continue on with the presentation. There's one more question that I'll just answer before I move on. So it says, it's again from Mark. It says, I know that DNA is double-stranded. How come the sequence is always shown in single linear form? I don't know exactly where you've seen the representation like this before. We'll obviously have a look at some of the ways that the genome and the DNA is presented in Ensembl.
Hello, sir.
I think.
Hello.
Hello.
Yeah, I've just seen in your slide where you represented why do we want to learn about Ensembl. In that slide, you listed.
You mean like this one?
No, yeah, something like this.
This one.
Usually they are represented this way instead of the double-stranded. I'm confused.
Yeah. So I think this is just it's convention, and it's because of the ease. Like it makes it easy to represent. So we know that DNA is double-stranded, but we also know that on the complementary strand, we know that A pairs with T and C pairs with G. So if we know the forward strand, if we know one strand, then we can work out the reverse strand, the other strand.
Okay, are we now assuming that the whole of this is just a strand? Yeah,
so if we're looking at this sequence here, for example, this might be the forward strand of just maybe a fraction of the genome. So it starts, say, C, G, G, C, C, T, T, T. But we know, and this is just, and then as it goes on to the next line, this is all part of the one strand of so like this goes A, T, C, A, C, and so it just goes on and on and on. But we know that if we were to look at the reverse strand, we could work it out here. So we'd know that on the reverse strand, this would be a G because it pairs with C. And then here there'd be a C because it pairs with the G on the forward strand. So we normally just give the one strand when we're reporting it, either the forward or the reverse, because that's where the feature is. And then we can work out the other one if we need to.
Okay, my question is this: what if in the other strand there is a mutation in the strand? How will we be able to identify? Assuming this is like this one you are showing now. Assuming this is a typical genomic sequence, and the complementary sequence, there's a mutation in that complementary sequence, the next sequence, the next strand.
Yeah.
How are we going to be able to identify that the other strand, there is a mutation in it?
Yeah. So, when you're looking at a variant or a mutation, they actually exist across both strands. So, for example, if this is the reference sequence, and here we have CAG, for example, you can see on my screen CAG. You would actually have, say, GTC in this section on the reference. But if this A was changed to a C, on the opposite, on the reverse strand, there'd be a G. So mutations and variants actually exist across both strands. So if there's a change in either the forward or the reverse, it will be reflected on the opposite strand as well. Yeah, and Dr. Sagan has actually also helped to answer this question as well. So this is sort of what I was trying to explain as well. So putting both strands will make it more confusing. If you want to do sequence analysis, for example, you can imagine how it will look if we have both strands. Yeah, it's sort of, we have sort of like scientific conventions, and we normally represent sequence in what we call FASTA format, where the header of the sequence indicates whether it's the forward or the reverse strand that is being reported and shown beneath, sort of thing. Okay, so I hope that answers the questions that you had. So I think what I'm going to do is I'm going to continue with the presentation, and then maybe Alina can answer some of the questions in the chat box. If there's time at the end, then I can go back and answer some of these questions as well.
Exactly.
Perfect. Good. So as I mentioned, when we are looking at Ensembl, we're looking at genes that have been annotated by either the automated or the manual gene annotation method. So just to think about these two methods in a bit more detail. So first of all, we have the automated gene annotation method. So this is a genome-wide determination. So I'm just going to mute everyone in the call. Okay, there we go. So the automated gene annotation is a genome-wide determination of the genes and transcripts using the Ensembl automated pipeline. And it makes predictions of the genome based on experimental data. It makes predictions of the gene and transcript models based on the data, the experimental data. So the type of data that's used by the automated pipeline as evidence is a range of different types of data. So first of all, we have what we call nucleotide sequences, which might include things like cDNAs, ESTs, express sequence tags, and RNA-seq data. This is all data that's stored in databases such as ENA, GenBank, and DDBJ. These are three different nucleotide sequence databases. So we take these sequences, the cDNA, EST, and RNA-seq data from these nucleotide sequence databases, and we feed that into the pipeline. The automated pipeline uses that and it aligns it to the reference genome to annotate the gene and the transcript models. The other type of data that the automated pipeline uses is protein sequences. So we take protein sequences from the SwissProt and Tremble databases, and we align those protein sequences to the genome and use that as evidence. The automated pipeline uses that as evidence to annotate the genes and the transcript models. The next sequence, the next type of evidence that the automated pipeline can use is the homologous sequence methodology. So we can infer genes based on homology and mapping between closely related species. So we can predict genes in the chimpanzee, for example, by mapping the human cDNA and protein sequences to the chimp genome. So even though there might be some slight differences, because the close evolutionary relationship between human and chimpanzee, we can use the human evidence aligned to the chimp genome to annotate the chimp genes and transcripts. As I mentioned, the other type of annotation is the manual gene annotation. So this is a gene determination on a case-by-case basis by an individual curator, a person sitting in the office assessing and analyzing the data. So they use a range of data. They use nucleotide sequences from the INSDC databases. They look at RNA-seq data sets. They also look at long-read transcriptomic data, PacBio and Oxford Nanopore, for example. They also look at mass spec data and also intron, CAGE, and poly-A-seq data. So a range of different data sets that the curators have access to, as well as the publications themselves, to also annotate the genes and transcripts. So this is what a gene and its transcripts looks like in Ensembl. So you can see that a gene is actually made up of a number of different transcripts. So in Ensembl, we call them transcripts. You might call them isoforms or splice variants. The terminology that we use in Ensembl is transcript. So what you can see here, for example, is the BRCA2 gene, which is actually made up of a number of different transcripts. So each of these different linear models is one transcript. So we can see here we've got one golden transcript at the top here. And the gold color, as you can see labeled on the right here, shows us that the gold color indicates that that transcript is a merged transcript. So that means that both the automated and the manual annotation methods have both independently come up with the same transcript model. So the same location and structure and size and exon and intron structure. Both of these methods have come up with the same model. So that gives us some confidence in the quality of the underlying annotation there. Then we have these other transcripts which are colored differently. So we have the red transcripts, which are protein coding transcripts. You can see that this gold transcript is also protein coding. So if it were not gold, it would be red. So red indicates all other protein coding transcripts that have been annotated by either the manual or the automated methodology. Then we have the blue transcripts, which are non-coding transcripts. So these might be things like process transcripts or nonsense-mediated decay transcripts. So there's no evidence of like functional protein products from these transcripts. If we look at one of these transcripts individually, we can see this golden transcript here, for example. You can see it's made up of these boxes and lines. So the boxes are the exons, like you can see here, and then the lines are the introns. When you're looking at the exons, the boxes, you can see that they're either colored in, like this here. So the colored in box is a coding exon, or you can see this box here is partly empty. So there's no color in this section of the box here. This indicates that it's part of a non-coding exon. So this is maybe the 3' UTR in this case. So this is the untranslated region of the sequence of the transcript. The final thing that I wanted to mention is about the Ensembl Ids that we have for our genes and transcripts and other features. So when we annotate a gene in Ensembl, we annotate it with we give it a stable ID, which always starts ENS, and then there's a single letter that indicates the type of feature, so G for gene, for example, and then a unique 11-digit number, which is unique and stable for that feature. And then we have the G is for genes, the T is for transcripts, the P is for peptide, E is for exon, and R for regulatory region. So all the different types of feature that we annotate in Ensembl have these this prefix before the unique ID code. This is important because in the lab, and when we're talking at conferences, and when we're talking with our lab mates, for example, we often talk about genes using their names, which is very easy and very, it fits into our language very nicely and they're memorable. However, gene names are not stable, and they're not standardized across different research groups and different communities. So genes can have different names and have synonyms, and different fields might use different terms for the same gene. And we do list gene names in Ensembl, but the stable and unambiguous way to refer to a gene is using this stable ID that we give you in Ensemble here. Good. So that brings me to the end of my first presentation. So what we're going to do now is move on to our first demo. So we're going to look at a gene called UQCRQ, and we're going to find information about that gene and some of the ways that we can view Ensemble and look at the data there. So you can obviously follow along with me, but if you go to the training materials, you can also see this is our demo that we're going to go through now, this overview. So if you open up a tab in your internet browser and go to www.ensemble.org, I'm just going to zoom in so you can see a bit more clearly. This takes you to the Ensemble homepage. So many of you might have been here before. I'll just spend a couple of minutes just explaining what we have on the page here. So at the top, we have this blue navigation bar. This appears at the top of every page, and you can use the links here then to continue your search or continue navigating around Ensemble if you need to. The logo is actually a link back to the homepage. So you can always use the logo to come back here and then begin your search again, or continue searching and looking through Ensemble. Over on the right-hand side, we have information about the different Ensemble releases and the new data that we've been incorporating into our Genome browser. On the main part of the page, though, you can see this search bar. So this search bar is your main way for searching Ensemble. So in this search bar, you can search for genomic coordinates, you can search for gene names, you can search for gene IDs, variant IDs, phenotype descriptions. There's lots of different things that you can search here. So if you go through the different modules, we'll see how this, you can see how the searching of different things brings you different features and different results in the results pages. Good. Underneath that, you can see that we have information about the different species that are available in Ensemble. So this is just a snapshot of some of the species we have, but if you want to see them all, you can click on View Full List of all species. And this takes you to a page. You can actually begin to see this table is really, really large. So you can actually see all of the different species that we have genome sequence and gene annotation for in Ensemble. You can obviously begin looking through this table and reading them for yourself, but you can also use the filter in the top right-hand corner here. So if I just begin typing Homo, for example, for Homo sapiens, you can see that my table is now filtered to show the Homo sapiens entry in the table. So we can see human is our species name, and then Homo sapiens is the scientific name. We've got the Ensemble Assembly. So this is the name of the genomic assembly that we have for human, GRCH38. And then this is the unique ID for the human genome assembly. What you can do to get more information about your species of interest, if you're interested in human data, for example, you can click on human in the table, and that takes you to the human homepage. So here we can see we've got a search bar at the top specifically for human, which we're going to use in a moment. And then underneath, you can see we have information about the genome assembly and then the different data types. So we have information about the gene annotation for human, for the variation data for human, the comparative genomics data for human, and the regulation data for human as well. So these are all the different data types that you can read more about and access as well through these pages. What we're going to do now is use the search bar at the top of the page to search for our gene of interest. So our gene is called UQCRQ. I'm going to just copy this and put it into the Zoom chat box so that you can all copy and paste that if you need to, or you can type it out yourself. And then once you've got your the name of the gene in the search bar, you can click Go. So that brings you to the results page. it's just like a Google results when you Google a different term. You can see these are all of our hits. So at the top, we have the human gene. So this is the UQCRQ gene. You can see human gene in brackets here. So if we click here, this takes us to a page that has information about the UQCRQ gene. So we covered this in the workshop in February. So I'm just going to quickly go through some of the information on this page, but I won't go through everything. So the first thing to notice is that you can see at the top, we've opened up these different tabs. So we searched for the UQCRQ gene. So you can see that we've now landed in this gene tab. So what we can see if we scroll down is information all relating to the UQCRQ gene. Over on the left-hand side, we have this menu. So this is a menu that shows us all of the different data about the UQCRQ gene. And you can see by default, we've just landed on this summary page. You can see summary is highlighted there in the menu. So when you look at the main part of the page, you can see this summary that shows you the gene name, the gene ID. Then you've got a description of the gene synonyms, the location of the gene and the strand that is annotated on. And then you also have this blue button that allows you to either show or to hide what we call the transcripts table. So if you click Show Transcript Table on the button there, you can find a table that shows you all of the different transcripts or isoforms, as you might call them, of the UQCRQ gene. So here we can see that we have 1, 2, 3, 4, 5, 6 transcripts of this gene. 3 of them are protein coding. You can see that in the biotype column. These 3 are protein coding. But then these 3 are non-coding. And you can see that because they've got this blue color, like I described in the presentation, this blue color. Then we've got links to other databases. So you can see here, for example, that we have links to the CCDS and the UniProt database. So if you want to get information about this protein, for example, you can click on the link here and it will take you out to the UniProt database. So this is where you might want to get information about the protein that this gene encodes. So you might want to get information about post-translational modification or protein expression or the protein structure and domains, for example. Obviously, this is a separate resource, so I'll let you explore UniProt by yourself, but we'll just go back to Ensemble now. Over on the right-hand side, you can see that we have what we call these flags. So these flags help us to prioritize the different transcripts. So when you're looking at these transcripts, your question might be, which one of these transcripts is the most biologically relevant and important transcripts for my study? And obviously, this is something that you need to think about for yourself in the context of your own research. So whether you're studying a particular disease or a particular tissue type, for example, it might be that your transcript of interest is different from someone else's. But these flags help you to begin looking at the qualities and assessing these transcripts. So you can see, for example, this top transcript is what we call the main select transcript. When a transcript is annotated as the main select transcript here, this means that the transcript is agreed upon between both the NCBI and Ensemble, and that it's highly expressed, clinically and biologically relevant transcript. So this is the main select transcript is often the transcript that you might want to prioritize when you're beginning your investigations. But obviously also remembering and appreciating that the other transcripts might be expressed under different conditions and different tissues at different developmental stages, for example, and that they also might have some biological role and might be important in your study. Good. As we scroll down the page, we can see that we have a graphical representation of the transcript. So again, just like I showed you in the presentation, we have these boxes and lines that show you the exon and the intron structure of the different transcripts. Good. So I won't show you everything, as I said, but over on the right-hand side, over on the menu on the left-hand side of the page, you can also see that you can access the sequence of the UQCRQ gene. You can access the ontologies, so the gene ontologies that give you information about the cellular component and the biological process that this gene is involved in regulating or is a part of. The phenotypes that this gene might be associated with, the genetic variants that are found within this gene, the gene expression pattern for this gene in different adult tissues, and then also the different metabolic and enzymatic pathways that this gene is involved in and the protein is involved in. And then finally, which I'll click on now, this link called external references is quite important. If you click on external references in the menu on the left, you'll just come to this page here that shows you links to other databases that have information about this gene. So you can see here, for example, this gene is found in Expression Atlas, where you can find information about expression patterns. You can see that this gene is also found in the MIM gene and MIM morbid databases that have information about phenotype and disease associations. We can see links to this gene in NCBI. So we've got this cross-reference between NCBI and Ensembl, and then also Reactome and Wikigene as well. So this is quite a nice place where you can come and cross-reference. So there's a question that's just come in that I'll try and answer now before I move on. So it comes from Akin Meiji. How can we get to know the gene names in Ensembl? So I guess there's not really a way to know the names. You have to sort of search for them. You can search using a number of different terms though. So if if it depends on your starting point. So if you've maybe done a literature review and you have the names of a few genes that you're interested in, you can just put the gene names into Ensembl, or maybe you've done some analysis and out of your analysis you have the gene ID, so you can search for the gene ID as well. If you don't know the names of your genes that you're interested in, but you're perhaps interested in a phenotype, then you can search for a phenotype such as type 2 diabetes. You can type that into the search bar and you can find the names of the associated genes and features that are linked to that phenotype. So it depends on your starting point and whether you've done a literature review or an analytical step, or whether you're searching for a phenotype. So it depends on what you want to do. Yes, you can also use gene synonyms as well. So you could, if you had a synonym like this, then you can also find that there as well through the main search bar. The final thing to mention to answer Akin Meiji's question is that there is a separate resource called genenames.org. Oh, it's not coming up for me. This is the consortium that's responsible for giving genes their names. So you can also come here and begin searching for gene names through here. And then once you have your gene name, you can come to Ensembl and search for your gene name as well.
What's the name of this?
HG. So this is the name of. I'll put the link in the chat box. This is genenames.org. So this is called HGNC, the Hugo Gene Nomenclature Committee. There's a final question that's just come in before we move on. It says that why is there only animal species are sequenced, not plant species? So at the moment, I'm showing you the Ensemble project, which is for animal or vertebrate species. But I'm going to show you a separate project that we have for non-vertebrate, including plant species as well. I'll do that at the end of this demo. So the final thing to notice is that you can, again, open this transcript table. You can also click on these individual transcripts. And when you click on these transcripts, you'll navigate to this Transcripts tab. So there's a new tab that's opened at the top here now that now shows us all of the information about this transcript. So the menu on the left has changed, and this is because we now have information about this transcript of this gene. The final tab that I wanted to show you is this Location tab. So this tab automatically opened when we searched for the UQ-CRQ gene. And this is the genomic location where the UQ-CRQ gene is found. So at the top, we have this chromosomal overview, and you can see this red box is our region of interest. And then as we scroll down, we begin to zoom in more and more on our region of interest. So this is our UQ-CRQ gene, and you can see that the data is now arranged into these tracks. So we have the contigs track, which represents the DNA. So this is these light and blue boxes represent the double-stranded DNA. And then we have the genes track here with each of these colored blocks representing one of the different genes. Then if we scroll down, the final view here you can see is what we call the region in detail view. And this is where you can see all of the different features in your region of interest. So we have this contigs track that represents the double-stranded DNA. And then we have different tracks showing us, for example, the genes with the UQ-CRQ genes and transcripts. And then we also have tracks showing us other types of features such as variants and regulatory features as well.
Please, how do you get to these, please?
Okay, so we clicked on location. So we were in the gene tab. This is where we were before. Hopefully you were also here as well. When you've searched for a gene, Ensembl will automatically open this location tab. So this is the genomic location where this gene exists. So we just have these different tabs for different types of features. So now we're in the location tab. So we're searching for the genomic location where the UQ-CRQ gene is found. So you just need to switch. You can switch between these different tabs at the top just by clicking on them to get these different views. Good. So the final thing that I want to do is show you how you can access the non-vertebrate species. So Effiong, there's also a question about parasites as well. So if you're interested in non-vertebrate species, then you can open up a new tab in your internet browser and type www.ensemblegenomes.org. So I'm going to copy this link and paste it into the chat box so everyone can click on it and see it. So this is our sister project. So it's very similar to Ensembl, but it's dedicated to non-vertebrate species. So you can see we have a section for Ensembl plants. We have a section for Ensembl metazoa. We have a section for Ensembl protists, protozoa. We have a section for Ensembl fungi and then Ensembl bacteria as well. So all you have to do is think about what type of species you're interested in. So if you're agricultural scientist, for example, and you're interested in plants, then you can click on the link here and it will take you to Ensembl plants. So what you'll be able to see is that even though the page is green, it's actually laid out in a very similar way to Ensembl, in the way that we have the search bar where you can search for the different features. You can search for your species of interest. You can search for your species of interest, just as we did in Ensembl as well. So yes, there's a couple of questions come in. So that brings me to the end of the presentation. So what I want to do now is give you a chance to have a go at the exercises. Before we do the exercises, there are a couple of questions that I can answer here. So the two that are very similar. So the first one is, is it possible to download a FASTA file from Ensembl to other programs for analysis? And then the other one is, now that we've searched for our gene, how can we navigate our way to get the nucleotide bases for both DNA or RNA of the protein sequences? So let's go back to our example here, the UQ-CRQ gene. So if you want to retrieve, so Mark and Akin Meji's questions are very similar about getting FASTA sequences. So if we click on sequence in the menu on the left-hand side, I'm just going to click on this button, hide transcript table, just so that the table's collapsed and we can see a bit more clearly here. So here now we can see the FASTA sequence of the UQ-CRQ gene. So you can see that the UQ-CRQ gene is highlighted in red here. So this is part of the sequence of the UQ-CRQ gene. This section up here that's got the black lettering, this is just upstream sequence that we show you by default, so 600 base pairs upstream. If you want to download the sequence, you can click on this blue download button. And here you can see, for example, it gives you all of the different options. So you've got the FASTA sequence.
Hello.
Hello.
Hello, Doug. Yeah, my own window is not showing same thing as yours. I don't know. My web page is not showing similar thing.
Did you click on sequence?
Yes, under the gene, UGCRG menu. I click on that and then hide the transcript table.
And did you click on sequence in the menu on the left-hand side?
Okay, no. Is it the secondary?
No, you it's just the sequence where my mouse is there. You just need to click on.
Okay,
there's no secondary structure because it's not an RNA.
Done, yeah.
Okay. So once you have the sequence, okay, so there's a question here. So what is the upstream sequence? So obviously what we're looking at here is just the sequence of the genome where the UQ-CRQ gene is found. So this first red letter here, this is the first base of our UQ-CRQ gene. This is just the upstream sequence. So if you have your gene, maybe your gene is located, maybe this is my finger, you just want the upstream, this is just the upstream sequence. So you could have 10 base pairs, 50 base pairs, 100 base pairs, 1,000 base pairs upstream. So you can just choose how much upstream sequence you want before the start of your gene of interest, which is given by the red letters here. So to download the sequence, as I mentioned, you just click on this download sequence button, and then you can choose if you want the genomic sequence, if you want the cDNA sequence of the different transcripts, if you want the amino acid sequence of the proteins that this gene encodes, or the exon sequence. So you can basically choose exactly what you want. So it depends upon what your research question is, or where you want to design primers, or all these sorts of things. So obviously we give you all the different options, and you need to think about what type of sequence you need to do what you need to do outside of Ensembl. So obviously once you've selected what you want, maybe the amino acid sequences in this case, you can just click download, and you'll see it's obviously downloaded onto my machine, but I won't do anything with that from here. You can obviously then work with it outside of Ensembl. Can we just copy our gene without the upstream sequence? So if you want to remove the upstream sequence, you have an option here. So there's this blue button on the left-hand side called Configure this page. So if you click on Configure this page, this will open up a pop-up window that allows you to manipulate the view of this sequence. So you can see here, by default, it's listed as 600 base pairs is the 5' flanking sequence, the upstream sequence. So you can just change this to 0, and you can change the downstream sequence to 0 as well if you want. And then you can click on the tick in the top right-hand corner, and then that will get rid of your upstream and downstream sequence if you don't want that as well. So you can use this configure this page button over on the left-hand side to manipulate the view and see exactly what you want to see. So you can choose if you don't want any upstream sequence, or you can change the length of the upstream sequence that you want to see. Good. Okay. So that brings me to the end of the demo. I think there's been quite a few questions, and hopefully I've answered them all. If there's any more, then obviously we can answer them throughout the exercise session as well. So what I want to do is maybe give you around 15 or 20 minutes to have a go at the exercises. So if you come to the training page, as we have done before, I can put that again in the link, the training page. If you click on Overview, and then find these overview exercises. So here we can see, for example, we have overview exercises, exploring the human MYH9 gene, finding a gene associated with a phenotype, and exploring a genomic region in human. So these are the different exercises that I would like you to have a go at doing now. Obviously, these are all exercises that focus around human. So if you want to have a go at looking at your own species of interest in the Ensembl plants or in Ensembl protozoa or Ensembl fungi, then please do take this opportunity to go across to those different resources and explore those as well, looking at your own species of interest. So it's now 10:15 here in the UK. So I think what we'll do is we'll come back together around 10:30, so in 15 minutes, and we'll see if there are any questions before we move on. If you do have any questions during the exercises, or if you get stuck, then please do ask a question in the living document or in the chat box, and Alina and I will do our best to help you.
Thank you so much.
So there's a question, someone's raised their hand. So Bamidele, I'm not sure if I've pronounced your name. Okay, so hopefully I didn't.
You tried.
I tried my best, yeah. How would you say this?
Bamidele.
Bamidele. Okay. Yeah, I'm sorry for my pronunciation. Yeah, obviously everyone else, please feel free to get on with the exercises, but I'm happy to answer some questions live as well. So yeah, please, Bamidele. Do you have a question?
Hello, that was a mistake. Sorry about that.
Okay. Can you lower your hand then? Because I think there might be some other questions as well.
I'm trying to.
Oh, I can lower your. Okay, I've lowered your hand for you.
Hello.
Hello.
Yeah, please, I want to be clear about these strands.
Yeah.
When we are talking about reverse strand and forward strand, what makes the difference?
I'm not sure. I'm not sure what you mean. Can you try and rephrase the question?
For instance, when we were searching the UQCQ gene,
yeah,
we noticed that it was located in a forward strand.
Yeah.
But in our exercise now, I'm noticing that it is located in the reverse strand.
Yeah.
I want to know what's the difference between reverse and forward strand.
Okay, so I'm gonna try and do some drawing for you so that you can. I can try. Like, I think it will be easier if I try and show you through a drawing on the screen. Okay. So hopefully you can see my drawing. I'm just going to do it over to the left-hand side. Can you see this drawing here?
Yeah.
Okay, so I'm just going to clear that and I'll start again. So DNA is double-stranded. as we've been saying. So we have maybe this is our forward strand, and this is our reverse strand. And we know that this is double-stranded, so we know that we have like an A here, for example, and this will pair with a T on this strand. And then we have maybe a C is the next base, and this pairs with a G.
Yeah.
Okay, sorry, I'm trying to do this with my mouse, so hopefully it's clear. So when we say that a gene or a feature is on the forward strand or on the reverse strand, what we actually mean is that the coding sequence of that gene is on the forward strand. So if a gene is on the forward strand, we would maybe label it like this. So this is an exon that we said. Oh, and then this is the intron. And then this is another exon. So we might say that this is a gene on the forward strand. So what that means is that the coding sequence of this gene is the sequence of the bases that we see on the forward strand. So maybe this is then T, C, G, A, T, C, G, like this, etc. Can you still see my screen?
Yeah, I can see.
Okay, so what that means is that when the coding sequence of this gene, okay, so when we see the coding sequence of this gene, this is C, T, G, G, etc. So this is that's when the gene's on the forward strand. But then what we know is that obviously on the opposite strand we have the complementary bases. So where this is T, this is an A, where this is a G, this is a C. This is a C, where this is a G, this is a C as well. So then when we say that there's a gene that's on the reverse strand, maybe if we forget about this one now, there's a gene on the forward strand, on the reverse strand, it means that the coding sequence of the gene are the letters, the order of bases that are on the reverse strand. So here it would be, because the reverse is obviously in this direction. Oh, the reverse is in this direction and the forward is in this direction. So the sequence here would be whatever's here, and then C, C, A, G, for example. So it's just when we say that a feature or a gene is on the forward strand or the reverse strand, we just mean the coding sequence of that gene is whether the coding sequence of the letters of the forward strand or of the reverse strand.
Okay, so I still want to be clarified on this. Does it really mean that forward strand implies a 5 prime, while the reverse strand implies the 3 prime?
Yeah, so forward strand is just, so this is the 5 prime end here on the left, and then this is the 3 prime end on the right hand side, like this. And then on the reverse strand, it means that this is the 5 prime, and then this is the 3 prime at this end on the reverse strand.
Okay, then is it possible that in each strand we can have different gene in each of the strands?
Yeah,
For
so
instance,
you can have
forward,
yeah, so
forward strand now.
Yeah,
what we
so
have from what you represent, sorry sir, from what you represented in the forward strand, assuming that is the gene for say VDR, vitamin D receptor protein, and then in the reverse strand we can assume that this is a gene for say vitamin A receptor protein or something of that nature, if there is.
So
is it possible that way, that is having 2 different genes in each strand?
Yeah, so basically, yeah, so obviously this is just a cartoon that I've drawn here, so it's not often that the genes are overlapping this much, but you can have genes that are closely located to one another or even overlapping on the forward and the reverse like this. So you can have some overlap between genes on the forward and reverse, yeah, for sure.
Okay. It's okay.
Okay, perfect. Okay, I don't know, my cartoon, I'm just looking at the quality of it. I don't just I've tried to obviously draw it with like a pencil on my screen, but it's not that clear. But hopefully it's the
No,
explanation
I
is good for you.
understand what you have done.
Okay, that's good.
I
I'll
understand.
get rid
The
of it
only
now.
area I will want to be clarified of is in the area of annotation. I'm just trying to get it clearly. What does it mean to annotate a gene or to annotate the?
So annotate, I guess we use the word annotate, but you could equally say that we predict. So we could say that we predict where the genes exist and the structure of the genes using evidence. So we say we predict. When you say the word predict, I guess it sort of people might assume that we sort of like guess, but actually we're annotating because we're using biological evidence to support the genes that we're predicting that region. So yeah, we use the protein and the nucleotide sequences to annotate where the genes exist. So we annotate their location, we annotate their size, we annotate the exon and the intron structure and the different transcripts. So it's just sort of like this, yeah, it's annotation methodology that we have. Perfect. Cool. I'm going to clear the cartoon now, and I'm going to.
Please, I have a question.
Yes.
Please, I want you to go back to that ensemble to show me where to locate the parasite, the plant. I'm trying to get it. I can't get it. Please, thank you.
Okay. Okay, so I think there's a few people that are unmuted. So who asked the question about the non-vertebrates? Was it Effiong?
Yes.
Okay, so I'm gonna mute everyone else. Just to get rid of the background noises. There we go. Okay. So if you want to find non-vertebrate species, then you need to open up a new tab in your browser and go to www.ensemblgenomes.org. And then that will take you to this page that has information about all of the non-vertebrate divisions that we have in Ensembl. So we've got Ensembl plants, Ensembl metazoa. So these are non-vertebrate metazoan species such as insects. Then we've got protists. So if you're studying a protozoa, you might want to come here. And then we've got a page also for fungi and for bacteria as well. So which one would be interesting to you, do you think, in this case?
Is the parasites.
So protists, yeah. So if we go to protists, you can see a number of different protozoan parasites here, for example, such as the Plasmodium. So this is obviously this is the malarial parasite.
Can we search? Is it possible to search for others? Because I'm interested in Schistosoma hematobium. Can I search? Where can I search for such?
Can you search for a specific species?
Yes.
Yeah, so if you click on this link here, view full list of all species.
Okay.
This shows you all the species, and then you can just begin typing the name in. I don't know the name of the species that you have there. So you can just begin, say, start typing Plasmodium, if that's, and then you can see here, these are all of the Plasmodium species that we have in Ensembl.
Okay.
Okay. Hello, Doc.
Hello.
Thank you very much.
That's no problem, Effiong.
Assuming I want to get something about Plasmodium now.
Yeah.
Am I going to get information on all the genes about Plasmodium, or just a specific segment of gene, or all the genes? Is it going to give me information on all the genes?
Well, it depends what you want to do. So you can see here, for example, maybe I want to search for this. This is the Plasmodium here, falciparum, which is the malaria parasite. So you can click here, and then we can search for specific genes. This obviously the genes in here, in the Plasmodium, they have this sort of ID structure like this. So you can search for a gene ID, or you can get information about all of the genes. If you click on more information statistics, you can just see the number of genes here, for example, over on the right-hand side. So it depends what you want to do. So you can either get one gene, or you can get you can export more genes as well if you want to, or like search for more genes if you want to. It depends what you want to do. Some people are just interested in one gene. Some people want to get information about all genes.
Hello, assume I want to get VDR gene on this Plasmodium. Is it possible?
VDR?
Yeah, VDR gene.
We can try and search for it. I'm not sure if it's... So VDR is not returning any search results. So in Plasmodium, they normally have these IDs like this, PF3D7, and then with a unique number. So it might be that you need to find the ID of this gene rather than VDR as the name.
Okay, can we search for NR111?
Well,
that's another.
I think there's some other questions. So I can leave you to try and search for the different genes yourself and see if you can find any, and then we can talk again later, maybe.
Okay.
Okay, perfect.
So, hello, Doc.
Sorry, I'm just writing there's a question from Adil in the chat box. So I'm just answering Adil, and then I'll come back to you in a second. Is that Akinmeji?
There is a question on the chat box.
Is this from Adil?
Yes, from Adil.
Okay, yeah. So Adil, I've just answered. We have a module later on this morning where we're going to be looking at how you can upload and analyze your own data to visualize it in Ensemble as well.
Yeah, hello, Doc.
Hi, Kimeji.
Yeah, sir. I'm just trying to, navigate my way on how to search for gene, the one you did first the other time. The UQR gene, I really right now to navigate my way through the interface.
You can't find it?
Yeah. So the first step in searching for it, the search bar pause and all that.
So let me just re-go over the steps for you then. So if we go to, we need to start at the Ensemble homepage like this. So I'll just put the link in the chat box for you. And then you've got a couple of different options. So firstly, if you know the species that you're working with, you can search for your species of interest. But if you just want to use the search bar from the homepage, you can do that as well. So you can just type UQCRQ. This is our gene that we searched for into the search bar. And again, I'll put that in the chat box for you. And then you can click go. And then you can click the result at the top from the search results. Like this. And then that takes you to the gene tab as we were exploring earlier.
Okay, Doc, assume we want to cite the information from this site. How are we going to do that?
If you want to cite Ensemble, you can click Help and Docs, and then over on the left-hand side, there's where is it? I can't see. Oh, it's scientific publications. Here we go. So we can see here there's information about citing Ensembl. So if you use Ensembl in your work, please cite the most recent overview article. So we have this Ensembl 2022, which is our latest paper that we have. So if you use Ensembl, you can cite this paper, and you can also, so you can cite this paper, and you can also, there are also other papers that reference more specific tools and data types if you want to also cite those as well.
I want to cite that specific page that listed the location of the gene and the information about the gene. Do I need to go through this process?
You don't normally need to cite specific pages. You can normally just cite the Ensembl overview article, like here. You don't need to cite specific pages. If you want to cite a page, you can normally just say you want to cite this page. You could just use the URL, the page address from your web browser. You could use this as your citation.
Okay. It's okay. Perfect.
Good. Okay. So there were some questions. It's now 10:35. So you've had around 20 minutes to have a go at the exercises. So what I think we'll do is we're now going to take a break. Can I do a recap? So we're going to take a break, and then we're going to come back again at 11 o'clock to move on to the next module. So I just want to give you a chance to take a break, stretch your legs, get some refreshments, and then come back, and we'll start again at 11, and we'll continue with the next modules of our course. So yeah, please do take the opportunity to go and take your break and then come back. We'll start again at 11, but Alina and I will both be here in case you do have any questions that you want to ask during the break.
Hello, good morning.
Morning.
Thank you very much for the session. I actually joined late. I thought it was going to be 9 a.m. I thought it was going to start by 10 a.m.
Okay.
I said I joined around 10. I joined around 10, so I thought the session was going to start at 10 a.m. So I didn't know it had started since 9 a.m. So I missed the better part of the training. I only came in to see all of this. So it's not like a magic to me. So I wouldn't mind if we can take the next maybe 5 to 10 minutes to like do a recap so that I can catch up on what has been said so far. Because even when I clicked on the link about the exercise, I just couldn't figure out what to do.
Okay.
So I don't intend to go for a break, so I wouldn't mind if you can please help me. Thank you.
Okay, yeah, so that's absolutely fine. There's someone else, Eric, who's asked for a short recap as well.
Yes, that is me.
Yeah, so I can, for everyone else, obviously, if you've been here from the beginning, then please feel free to go and have a break. But for people who did join a bit late, I can very, very quickly just show you a couple of things that we were doing. And then we can all come back again at 11 and continue from that point. So as Alina said in the chat, this is important to notice, is that we are recording the session. So if you did miss stuff at the beginning, then after the workshop today, I'm going to process the recording and I'll send it around to Ashraf, who can circulate it to you all as well. So you can watch the recording as well in case you want to re-watch that and catch up on anything that you missed. So For those people who do want a very quick 5-minute recap, what we were doing is we were thinking about Ensembl and the data that we have here in the Genome browser. So if you've never used Ensembl before, it's important to notice that we have these 4 main types of data. So we've got the Genomic Assemblies, which is the genome sequences of a number of different species, and then the gene annotation of those genome assemblies. So Ensembl's not a sequencing project. We don't sequence the genomes of different species, but we take those genome assemblies that have been deposited into publicly available archives, and then we annotate them using an automated gene annotation pipeline. So that's the first type of data. And that's all we've really thought about so far this morning. Then we also have the variation, comparative genomics, and regulation data. So these are the 4 main types of data in Ensembl. But as I said, the only thing that we've really thought about in any detail this morning is the genomic assemblies and the gene annotation. So the main thing that we did was we sort of went through Ensembl, and we saw how we could search for a gene of interest. So if you go to www.ensemble.org, you can begin searching. So I've just put the link in the chat box. You can begin searching for genes of interest. So we search for a gene called UQCRQ, but you can search for anything you like here. So gene names, gene IDs, maybe BRCA2 is your gene of interest, maybe UQCRQ is your gene of interest. Obviously, I don't know exactly what you're working on or what you're, which species or which genes you're interested in, but just as an example, you can search for UQCRQ. And then this takes you to the home page, the results page, where you can then see this is our search results. So the human gene is listed at the top here. So we can click through, and that takes us to the UQCRQ gene tab, where we can explore information about the UQCRQ gene, its sequence, its function, functional data, expression data, cross references to other databases, and everything as well. That's really what we've been mainly doing. So if you want to see the full walkthrough of what we did, this is actually all written out for us here. So these are our training materials for the course today. So if you follow that link, you'll find a page here that has information about the workshop, and then we have the modules at the bottom here. So we have overview, regulation, and custom data. So these are our 3 modules today. So in the overview section, this is what we covered this morning. So we've got the overview demo, which just tells you about the different pages that are there on Ensembl and how we can search for different genes. And then the exercises as well here. So please, I think the best thing to do is to have a little look at this demo. Maybe try and go through the demo by using the instructions and the descriptions that we have here for you. And then that will take you through what we covered in the module. So for Mary and Eric, hopefully that's enough to get you started.
All right, thank you so much. Let me quickly do that now, and I'll get back to you, sir.
Okay, perfect. No problem.
All right, sir. Thank you very much. I still have a question, sir.
Okay.
Please, I'm working on a particular parasite, and his name is Schistosoma hematobium. I don't know if you've heard of it before.
No.
Okay, Schistosoma hematobium. So I want to find out if I'm to design a primer that can be specific, how do I go about it? I don't know. I know this is not a primer design class, but can you put me through, or should I type the name of the organism for you to help me search for some things?
Yeah, so is it schistosoma?
Hematobium.
Like this?
That is it. Yes, okay. That is it. Yes.
So we can, I'm not sure if it's present in Ensembl.
it's not there.
Okay. So basically, if your species is not
NCBI.
Pardon?
Checking on NCBI's webpage, you can see something on that.
Yeah, so it basically, if there's not a, it's not always the case that if a species has its genome sequence, then it's present in Ensembl. So if the genome of the schistosoma that you're studying is not in Ensembl, then you might need to just, but it is in NCBI, then you might need to just use the NCBI resources instead.
Okay. Okay. Okay.
Okay.
All right. Thank you, sir.
I have a question.
That's okay. Yes,
I have a question,
please.
sir.
Yeah.
Yeah, I was wondering whether it would be possible for one to get all the necessary information with respect to a particular publication on Ensembl, and then without incorporating other bioinformatic tools. For instance, I want to work on malaria parasitemia, and I want to look at the protein. I want to visualize it and give information to people. Will it be possible without going to getting other bioinformatic tools into play?
Well, I guess I mean, Ensembl is one resource that we have. So Ensembl is one resource that covers information about genomes and genes and variants and things. So Ensembl is a very wide resource. It does have a lot of data, but I think that the best thing to do is actually to use Ensembl alongside all the other different databases as well. So firstly, it depends on what data you're interested in. You might only be interested in genes and genomes, but if you're also interested in proteins, then I would really advise also looking in databases like UniProt and PDBE that have information about the proteins and the protein structures. So all the different databases have different focuses and they look at different types of biological data. So if you want to really get a full picture about even the proteins and the protein function and structure, then it's probably best to try and use different databases alongside one another to build up a bigger picture that you can. Because Ensembl is going to have some data, but it won't have information about it won't have detailed information about proteins and protein structure, for example.
All right, please, I still have one more question, sir.
Okay, so sorry, I'm just trying to organize. So I think that was just Mark's question. So I'm just going to, I can mute Mark. I think Eric, is it your? Did you say you had another question?
Yes, okay. Yes, I have a question.
Yes, yeah. Okay. So we have Eric, and then I'll move on to, I think, Effiong. You have another question as well.
Yes, sir.
Okay, so let's start with Eric, and then we'll move on to Effiong.
All right, thank you so much.
That's okay.
I joined late. I was trying to follow up on the recap you just gave some 5 minutes ago.
Yeah.
But I have seen these annotations on the NCBI. Is it the same with the annotations Ensemble does, or is this slightly different? Because the annotations I saw various SNPs on NCBI. I saw that on NCBI using their variation sequencer 3.212 thereabout. That's the software they use on NCBI.
Yeah.
To get the annotations on gene. Is it the same thing as what Ensemble does, or different?
So the important thing to think about at the beginning is the fact. So NCBI and Ensembl are two different resources. So NCBI is sort of organized and maintained by groups in the US, whereas Ensembl is hosted and maintained here in Cambridge in the UK, in Europe. So the data that you can find in these two different databases is often the same. So the underlying data is the same. So if you're looking at the human genome, then the human genome sequence will be the same in Ensembl and in NCBI, because they're they're both using the same underlying genome sequence for human. But then the differences come in because there's different annotation methodologies. So Ensembl might annotate genes using a particular method A, for example, whereas NCBI has a slightly different gene annotation methodology. So sometimes there's some slight differences in the gene structure and name, and so they often do exactly match, but there can be differences as well. So this is always important to remember when you're using Ensembl or NCBI, that you can look at them both and try and cross-reference and like build up a picture using information from both databases, but there's not always a one-to-one exact mapping between all of the annotation in both databases.
Okay. And also, let me also ask, is it possible to just do this dry lab, like without going to the lab, just on the website and use it in conjunction with other bioinformatic tools and publish papers?
Yes. I mean, yeah, there's many people that do purely bioinformatics sort of dry lab projects, and they sort of do all of their analysis through public data sets, and many people sort of collaborate with wet lab groups, and they get data in that way as well. So it really depends upon your project and what data sets are already available, and whether you can use data sets that have been published and are publicly available. It's very dependent, and it's dependent on the context and what project you want to do and what data sets are already there for you to use.
Okay, thank you so much.
Yeah, no problem. Effiong, we'll move on to you. Yeah.
All right, thank you very much. Please, there are some terminologies I want you to put me through, like annotation, open reading frame, context, read. These terminologies, I don't know how you could please explain to my understanding.
Okay, so the first term that you're not sure on is annotation?
Yes.
Okay, so when I say annotation, I basically, I mean the prediction of where the genes exist in the genome. But when I say predict, I don't mean guess, I mean use underlying data to annotate that. So if I'm looking at a region of the genome, it doesn't really, I mean, say I'm looking at a region on chromosome 5, and I can see, okay, I've got a cDNA sequence and a protein sequence, and they both align to this exact region in the genome, then I can use those nucleotide and protein sequences as evidence that there is a gene at this position in the genome. And I can even use that evidence to predict the structure. So I can say that this part of the genome is encoding an exon, and this part of a genome is the intron, and then this next part is an exon again. So I basically use the underlying evidence, so nucleotide and protein sequences, to predict and label where the different features, such as the genes, exist in the genome and what their structure is. That's what I mean by annotation.
Okay. Please, what about open reading frame?
So open reading frame. So in Ensembl, when we do this gene annotation that I just sort of mentioned, we annotate based on this evidence. So it might be that we use all these different nucleotide and protein and different data sets to predict and to annotate the gene structure and location. However, another way that you can look for genes and to annotate genes as well is by searching for open reading frames. So we know that the first translated codon of a transcript of a gene is ATG. Okay. So this is the triplet codon that encodes the methionine that is the first residue of the protein. It's the first. The ATG is the first part of the translated sequence of the transcript. So one thing that you can do if you're trying to annotate genes is that you can search across the genome and find these ATG sequences.
Okay.
And then try and use those to predict where like the genes start, sort of thing. So it's not a very accurate method of predicting where genes exist, because there are ATGs that don't encode the start of a gene, or so they, it's not using any underlying evidence. It's just searching for this pattern of sequence, the ATGs. Whereas what we do in Ensemble is we use this evidence, such as the protein and the nucleotide sequences, to say this is the gene here and this is its sequence.
Okay, please, what about contigs?
Okay, contig. So contig it's a technical term that is used for describing how the genome was broken down into pieces during its sequencing. So, for example, maybe I can draw again on my screen. It will be easier to do. Okay, so I'm just going to draw at the bottom on my screen here. So say you've got your genome like this. Well, so it's not a straight line, but you can see what I'm trying to draw here.
Yes.
So when we sequence the genome, we can't sequence the whole thing because the technology hasn't got the capacity to do such long sequencing. So what we do is we break it into these chunks like this, and we sequence the chunks, and then we put the chunks back together. So these are what we call, this is a oh, sorry, my pen's gone. This is what we call here, this is a contig, and then this is the next contig. So the contig, it's like a puzzle. So it's where you've broken the piece, the puzzle's been broken apart into the individual pieces, and then we put the pieces back together to get the final picture. So each contig is a puzzle piece, and then we put the puzzle pieces back together. Obviously, when it's a genome in a linear fashion, rather than like a picture is 2D, whereas DNA is one-dimensional, just along like a straight line. So we have these contigs, which are the puzzle pieces where we put them back together.
Okay. So this contigs is what we now assemble together to form the whole sequence.
Yeah, exactly. Yeah.
Okay. So what about the reads? The reads. How do you explain reads?
So reads. Well, this is more to do with the technology behind doing the sequencing of the genome. Let me just get rid of this picture. And this is behind the technology behind the sequencing. So when you are sequencing a sample, you don't just get one sequence, you get lots of different. So this is the genome, for example, here. You might sequence a fragment like this, and 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 this, and this, and this. So this is these are all different reads, which might be sort of 50 to 200 base pairs in length. And then, so what we do is we make the contig like this. Oh, we make the contig like this underneath for this region. This is the contig here, by matching up these reads that are overlapping.
Okay.
So you can see, for example, maybe this read has got a C at this position, and so is this read, and so is this read. So these are all individual small sequences that, as a consensus, give us the C at this position in the contig here.
Okay.
So these are all individual reads.
Okay. So from what you explained, it looks like read is, okay, it's like when we say set, subset, the read is the subset of the contig, right?
Well, not a subset. They're actually the raw data that's used to construct the contig.
Okay. But how about in the process of assembly? Don't you think there could be error picking the wrong? Maybe this one came first, this one came second, and is the system so perfect to pick the rate accordingly when assembling?
Yeah, no, that's a very good question, and you are right. Like it's not a perfect process, and in fact, when you're looking at the genomes of different species, then there can be errors and there can be gaps in the genome sequence. And this is just where the technology is not good enough to sequence that region of the genome. Maybe there's a really highly repetitive region in the genome, or there's some technical difficulties associated with that part of the sequence. So yeah, there are errors, and what we do over time is we release, well, when I say I mean like the scientific community. So the scientific community will resequence the genomes of different species as the technology improves to try and to correct those errors and those gaps and fill them in.
No, that's a very good one. Okay, how do we get reference sequence? Because for a sequence to be called reference, that means it has been approved like standard. So how do we ensure the perfection in reference sequence, or do we keep trying reference sequence over and over to perfect it?
Yeah, so no, so the reference sequence just means, so it's just, the reference is just actually the current best sequence that we have. So it can be that, you know, so when the human genome was first published in 2004, there were lots of gaps and lots of errors, but it was the reference because it was the best that we had at the time. And then obviously over the years, there's been more sequencing has been done and more advanced technologies have improved what we have. So the reference is updated over time. So we now have a more complete and more accurate reference.
Okay, because why I'm asking this question, some of the reference we just go to NCBI or Ensemble to pick, and there are errors, and we use it to, you know, publish and make judgment, and maybe you also use it in drug design. Don't you think when those references are not correct or not properly sequenced, it can affect the drug design that we go about doing docking, and what the implication of that on human being when we are not really sure of the sequences that are being deposited in the gene bank?
Yeah, I mean, it's just it's something that as scientists we have to always be aware of, and that we have to be prepared that we have to look at lots of different lines of evidence and try and ensure that when you're doing a wet lab experiment, you always have to look at error bars and make sure that, you know, you've got obviously human error and biological variants and stuff within your samples and within your replicates. So this is something that we always have to be thinking about, and when we're aware of when we're doing experiments and designing experiments. So as in with the wet lab, we just always have to be aware of these features, these things when we're doing our science. Okay. So
thank you so much.
That's okay. That's okay. It's now 11 o'clock. So I hope that, obviously I've been answering some questions. I hope that other people have had a chance to also do some exercises and to have a break. There's a question. So Mary has written, you said that you've got some questions. So
yes, should I please say just in a few minutes?
Yeah, I think the best thing is if you have a couple of short questions, then we can answer those and then we'll move on. If anyone has any longer questions, then I suggest that you put them in the chat box, and then Alina and myself can answer them like as we progress through the course today.
You've not had a break yourself.
No, I know, that's okay. I can take a break at the end. So let's Mary, if you have a couple of questions, and then we'll move on. Yeah.
All right, thank you. I was able to actually maximize that environment you said, and it was quite interesting. But I want to ask just a few questions. Is it possible to encounter missing sequences as we explore that interface? And then I just want to quickly answer. If it is a positive answer, then how can these missing sequences be built? And also, what's the significance of this ensemble? So is it used for drug design, drug discovery, and all? Because I know that for this bioinformatics, we have different aspects. There's the part of drug discovery, there's part of drug design, there's a part of drug development. So I need clarity on that. Thank you very much.
Okay, so I'll try and answer some of the questions. Hopefully, yeah, there's a few questions that you asked there. So the first one, I'll answer your last question first. So you wanted to know about where Ensembl fitted in terms of drug design and drug discovery and other types of research as well. So I think the important thing to remember is that Ensembl is a genome browser. So we have information about genome sequences of different species, and then we annotate a range of different data types. So the main type of data are the genes, but then we also have information about variants and phenotype associations. We also have some comparative genomics where we look at homology relationships between species. Maybe people are interested in that if they're studying evolution. And then we also have information about regulation of genes, for example. So we have a wide range of data that are, but really only relating to genes and genomes. So if you're interested in drug design and drug discovery, for example, then you might be interested in Ensembl, but you're also then going to want to take what you learn in Ensembl and then also look at other databases such as protein structures and protein informations from UniProt or PDBE. Because obviously knowing information about the gene can tell you perhaps about some expression levels or the structure of the gene or where, like, the sequence of the underlying DNA. But if you're designing a drug, then you want to know a bit more information about the protein and then the structure of the protein and where different residues are in 3D space. So then Ensembl doesn't have so much information about this. So you might want to go to another database like UniProt or PDBE that does have information about that. So I think it's important to always to appreciate Ensembl and to know about the data that it has, but to also know that there are other databases that have information about other types of biological data, such as proteins or chemical entities, or those sorts of things.
All right, thank you very much, sir.
That's okay. Yeah, if you have any other questions, Mary, then please just pop them in the chat box.
Yeah, okay, please, before we go, I want you to throw more, to shed more light on ligands. What do you really understand by ligands as pertaining to docking? What do you really need more explanation?
Okay, we don't have any information about molecular docking and ligands in Ensembl. So you need to, I think at the very end of the workshop today, what I'm going to do is I'm going to show you some of the other databases and resources that we have in EBI that have information about proteins and dockings and ligands.
Okay.
Perfect.
All right. Thank you, sir.
So I'm going to mute everyone now, and I think we're going to move on to the next module.
All right, sir.
Okay. Oh, sorry. I just need to. So the next module that I wanted to look at is called custom data. So some people have already asked this question before in the workshop today, and it's about how you can visualize your own data in Ensembl. So what we've looked at already so far is how you can look at the publicly available data in Ensembl. So when you go to the Ensembl website, the data that you're looking at is free for everyone around the world to look at and to analyze and to view on their own web, on their internet, on their own computer. However, what you can do is you can upload your own data to Ensembl to visualize it privately against all of the publicly available data and begin analyzing and assessing your own data sets. So Ensembl supports a number of different data types. So I don't expect you to know or to remember any of these data types off the top of your head. Maybe you are familiar with one or two of them, but these are the different file formats that Ensembl supports. So BAM files, GTF and GFF files, BED and BED graph files, BIGWIG, PSL, VCF, and pairwise interaction. So these are the different file formats that we support. We're going to go through some of them in a bit more detail in the module today, and we're going to see how we can upload them to visualize them in Ensembl. So if you want to attach your own data, you can upload data directly, or you can attach files remotely. So there's a 20 megabyte, sorry, I'm just going to, there's some background noise, so I'm just going to mute people so that there's a 20 megabyte limit on the size of the files that you can upload directly. So if you have a really large file, then you'll need to attach your file remotely using a URL based methodology that I can show you later on as well. So if your file is more than 20 megabytes and you need to attach it remotely, as I mentioned, you can create a URL by using either your institute's FTP site, if your institute has one of those, or you can use one of these freely available platforms such as Cyverse or Figshare that will allow you to create a URL where your data is saved and then to upload that to Ensembl. So what we're going to do in this hands-on presentation is we're going to upload what we call a bed file. This is a bed file that contains description of these genomic regions. So we're going to imagine that these are deletions from patients that have been sequenced, and those patients have microcephaly and developmental delay. So these are 3 different patients that have all presented with similar symptoms, and we through performing some sequencing of their genome and their exomes, we have found that they all have these they each have a large scale deletion. So this first row of this data is showing us the deletion in patient 1. So we can see that the first section of this data is telling us that we're looking at chromosome 5. Then we have a gap, then we have our start coordinate, then we have a gap, then our end coordinate. So these are the genomic coordinates that are deleted in patient 1. So everything between these 2 coordinates is deleted in patient 1. This next row of data is for patient 2. So you can see here, for example, that they also have a deletion on chromosome 5, and the deletion is between these 2 coordinates. So this is the start of the deletion, and this is the end of the deletion. And the same for patient 3. They also have a deletion on chromosome 5 between these 2 coordinates, the start and the end. So the question here is that's coming in from Ayodele is, what is the meaning of the bed? So bed, I'm just going to say, if you are on the call, then please do make sure that you're muted, because otherwise it interferes the background noise with background noise for everyone. So a bed file is just a file that contains information about a genomic feature, and it just gives you that as the genomic coordinates. So this here that's on the screen highlighted in red, this is a bed file. So a bed file always has these particular columns. So it has the chromosome, the start coordinate of the feature, the end coordinate of the feature, and then an optional descriptive term here. So we've just added P1 as the description here, because we know, then we can know that this relates to patient 1. And then the next row of the data is for the next patient. So each row of the data, each row of the file refers to the different features. So this is 1 deletion, this is the next deletion, and this is the 3rd deletion. Good. Okay. So if you want to follow along with me, we're going to open up firstly open up the materials for the workshop. And we're going to click on this module here called Custom Data. So if you open up the demo, I can share the link with you in the chat box again. So you can see here, this is the bed file that I was describing. And then underneath, this is the bed file itself. So you can see we just have, it's a small bed file. Sometimes bed files are really, really big files with many rows because it contains information about lots of different features. But here we just have 3 features, these 3 deletions. So we can copy this from the walkthrough that we have here on the page, and we can paste this into Ensembl when we are doing our demo. So if we open up a tab in our internet browser and type www.ensemble.org, the first thing that we can do is we can search for our species of interest. So we know that we are looking at deletions that have been identified in clinical settings, so human patients, obviously, that we're looking at. So we can click on View Full List of All Species, and then use the filter in the top right-hand corner to search for Homo sapiens, and then navigate to the human homepage just as we did before. So what we can do now is you can see that in this section in the top left-hand corner, it's called Genome Assembly. There's a link here that says display your data in Ensembl. So if you click here, display your data in Ensembl, this will give you an option for uploading your data. So what we can do in this case is we can give our data a name. So maybe we'll just call this, I'll call this one Noblekinmat, because this is the host for our workshop today. But obviously, when you're uploading your own files, then you might give the data a more descriptive name to let you know what it is. And then we can upload our data in this box. So what we're going to do is we're just going to paste our bed file directly into the text box. But you could also upload a file if you have when you're using Ensembl in the future. If you have a file saved directly on your computer, you can upload it by clicking on the option here and selecting the file from your computer, or you can provide a URL into here as well. So if you have your file saved as a URL, something like this, if this is your file, for example, then you can put the URL in here. But we can just copy and paste from our example. The final thing that we have to do is to choose our data format so that we know that our file is in bed. So we can select bed file format from here. Once you've selected the file format, you can click Add Data, the blue button at the bottom. And you can see in the pop-up window now, it says, thank you, your file has been uploaded successfully. So we can go to it says go to the location view with the nearest region with our data. So we can click on the link here in the pop-up window. And this takes us to the location tab. Okay, so there's a couple of people who've, Ayodele and Elizabeth have both asked to go back a step to see how we get here. So absolutely. So if you're on the Ensembl homepage, the easiest thing to do is to search for your species of interest. So from the homepage, click on this blue link that says View Full List of All Species. And from here, in the filter, in the top right-hand corner, you can search for your species of interest. So I'm going to type Homo, H-O-M-O, to begin searching for Homo sapiens. And you can see that my table is now filtered to show my species of interest. So I click on human, And that takes me to a species-specific homepage. So from my species-specific homepage, I can see that there's this section in the top left-hand corner here. The title is called Genome Assembly. You can see that's the title of this section. And underneath there's a link that says display your data in Ensembl. So that's where you need to click there to upload your data. And then that brings up the pop-up window that we've been looking at just now that allows you to actually submit your data and to add it. So you just go through, you add your name of your data, you paste in your data from the example, and then you choose the file format. So we have a bed file format, so we need to select bed from the drop-down list there. And then you click add data. So hopefully that everyone's on the same place now. So what we can do is we click on the link in the pop-up window that's asking us if we want to go to the region in the genome where our features are located. So we can click here, and this takes us to the location tab in Ensembl. So we looked at this very briefly earlier on in the last session. So here we can see we're in the location tab, and we're in this section called region in detail. So here we can see this is the page with all of our information. This is the chromosomal overview. You can see this is the chromosome. So this is the P arm of the chromosome. This is the centromere here. And then this is the Q arm of the chromosome. And then we have our red box. You can see the red box hopefully on your screen as well. This is our region of interest. So this is where our data is located on the genome, the three features that we had in our bed file. And then they will be displayed on the images further down the page. So the next image here is, again, what we saw earlier, what we call the region overview. So this is just a 1 megabase overview that shows you your region of interest, which is, again, highlighted by this red box. So this red box is our region of interest. And then we have data in these different tracks. So we have the contigs track. As we mentioned earlier, the contigs track is representing the double-stranded DNA. So this blue bar is alternating light and dark blue as we move along here. This blue bar represents the double-stranded DNA. And then we have data in these other tracks. So we have the genes track underneath that shows us all of the genes annotated in the region, all aligned to the genome, the double-stranded DNA there. If we scroll down, we can now see in detail, this is our region of interest. So this is, this first track here that you can see is highlighted. This is the one called Noblekinmat, and this is our 3 deletions that you can see. So we've got P1, P2, and P3. So these blocks represent the region of the genome that has been deleted in these 3 patients. So if we scroll down, we can begin to actually see which features are affected by these deletions. So what I'm going to do is I'm going to go up, and I'm just going to use this plus and minus icon here to zoom out. So if I click on the minus icon, I'm just going to zoom out a little bit more so that I can actually see the boundaries of my deletions. The page is just reloading for a minute. Okay, the page is just taking a little bit longer than expected, so I'm just going to give it a minute or two more just to allow the page to reload. Okay, so I'm not sure what's I'm just going to try reloading the page again. Are other people encountering this same waiting icon, the loading icon?
Hello, actually, I'm not practicing because I don't know where you got the P1, P2, P3 sequence from. If I know, I would have joined, but I don't know where to get it from.
Okay, so you can get it from here. So if you go to the materials for the course today, I've just put the link in the chat box. Then you can copy these. You can copy the data and use it to
Yes,
have a
how
go
do I
yourself.
get this data? Yes, where do I get the data? How to get this chromosome 5, P1, P2? If I can get the data, then I'll join in what you're doing.
Yeah, well, so we're obviously just we're using this as an example. We're just this is just a small example data set. So these are 3 deletions that have been identified in 3 different patients. So it's just an example, but you might have generated data in another way in your lab, for example.
Or how do I have a question, please.
Yes.
If I have a sequence, how do I convert it to a bed file?
If you have the sequence.
Yes.
So you have maybe like just.
I've generated data in a wet lab, and I want to convert it now to a bed file or any of the file format accepted by Ensemble.
Yeah. So you have to maybe, I mean, you won't be able to do that with Ensemble. You'll have to use another platform. So you might have to do some alignments. So you can, so you can, for example, maybe using something like Blast or some other alignment tool, you can search.
Yes, I've done all that. I've done all that.
Yeah. Okay, so then if you, I mean, you might be able to Google how you can convert like a FASTA file or a set of coordinates into a bed file. If you've got, it depends if you've got the coordinates or if you have just the sequence still.
Okay. Apart from the sequence, I need to get the coordinates as well too.
Yeah. So the bed file specifically has the coordinates. So that's what a bed file contains. So it contains the coordinates. So you need the chromosome number, and then you need the start coordinate and then the end coordinate. So for example, if you're looking at a deletion, then you specifically need to know where the deletion starts, the genomic coordinate where the deletion starts, and the genomic coordinate where the deletion ends as well. So this is what the file contains. So it has got the chromosome number, and then the start coordinate, and then the end coordinate. So these are the 3 pieces of information that you need for each of the features that you want to look at in the genome.
So that means I cannot upload a FASTA format to the Ensemble website.
Yeah, so Ensemble doesn't support FASTA as an upload format. So you can obviously visualize the genome sequences, but you can't upload your own FASTA sequences to visualize in Ensemble. You need to convert them to bed file format or something like that, or another supported format. Okay.
Please, I have a question.
Yes, please.
I want to ask. I already went to the material, so and I already copied, but I'm on the site. I don't know how we got to that where we pasted the BAM files.
Okay, so if you just, I think we did this a couple of times a bit earlier. So if you go to Ensemble.
Yes, I'm in this interface now.
Yeah, and then if you search for your species of interest, so in this case it's human, because our example data comes from a clinical setting, so it's human data. So we click on View Full List of all species, then click on Filter and begin typing Homo sapiens. This is our species of interest, then click on Human. So hopefully you'll hear us now.
Okay.
And then
Let me try it out.
From here, you then need to click on this link that says display your data in Ensemble.
Okay.
And then you can upload your data in this pop-up window.
Okay, thank you. I will try it out.
Okay, so my page now has, so from where we left off before, my page is now reloaded, and you can see that we have the data is all aligned to the genome. So you can see if we scroll down, if we scroll down a long way, you can see that we have what we call the contigs track. So you can see here, this contigs track, as I said before, again, this represents the double-stranded DNA. And then the data in all of the other tracks is just aligned to this genome sequence, this DNA, this double-stranded DNA. So you can see, for example, we have all of these transcripts of the SLC1A3 gene. This gene has got lots of different transcripts. You can see them all annotated here. And we can also see at this end, further downstream, we also have transcripts of another gene called the NIPBL gene, NIPBL. So this is another gene, and this has got these transcripts annotated here. Then we have some other data tracks, which you can obviously click on and learn about if you're interested in these other data types. But what we are mainly interested in are these 3 deletions that we uploaded. So we've got P1, P2, and P3. So we can see that P2 obviously deletes this region of the genome. So if I click and drag, you can see that P2 is deleting this whole region that I've highlighted. So it's actually going to delete a big portion of this NIPBL gene. So the first perhaps 1, 2, 3, 4, 5, 6, 7 exons of this gene are all going to be deleted by the deletion that we observe in the P2 patient. When we look at P1, we can see that, in fact, even more of the NIPBL gene, in fact, all of it is being deleted by the deletion that we see in this P1. And then P3 is actually a smaller deletion. So it's not deleting the whole of the NIPBL gene. It's just leaving this first exon, but the majority of the NIPBL gene is all being deleted in this case here. So all of this highlighted region is all being deleted. So this is a way that you can obviously visualize your data, and now you can begin to understand, okay, these 3 patients, they all came to the clinic, they all presented with the same symptoms, and when we sequenced their genomes, we identified these 3 different deletions, but they actually all affect this same gene. So what we can do, maybe we see that these genes are all affected by the same deletion. This one gene is affected by all these 3 different deletions. So we can click on the gene. So if you click on the gene from the view, I'm just waiting for the pop-up window to open now. So when the pop-up window loads, you can actually use the links here to find out more information about the gene. Well, you can read more information about the gene in the pop-up window, but you can also just click on the links to go to the gene tab and find out more information about this gene. So if we click on ENSG, the ID for this gene in the pop-up window, we can now read more information about this NIPBL gene, the NIPBL gene. So this is the page that we saw before, the gene tab. If we scroll down, you can see that there's a section in the menu on the left-hand side called phenotypes. So what we might like to do in this case is to see whether the NIPBL gene has got any known associations with the phenotypes that we've observed in our patients. So we can click on phenotypes. And indeed, you can see that this gene is associated with this 5p13 microduplication syndrome, but also Cornelia de Lange syndrome. So if you want to read more information about this Cornelia de Lange syndrome, you can click on the link for the source. So MIM-Morbid is the database that has information about the Cornelia de Lange syndrome. And if you click there, it takes you to the OMIM website that has information about this disease. And you can see that the Cornelia de Lange syndrome is a multi-system malformation syndrome recognized primarily on the basis of characteristic facial dysmorphism, including low anterior hairline, arched eyebrows, and a number of other facial characteristics. And this is exactly what we saw in our 3 patients. So what we can now say is sort of, well, this is not a diagnostic test, obviously. We can't perform any diagnoses using Ensembl, but we can begin to hypothesize that perhaps the deletions that we observed in the patients are affecting this NIPBL gene, and we know that the NIP through searching Ensembl, we know that the NIPBL gene is already associated with other patients that have this syndrome, the Cornelia de Lange syndrome, which is mirroring exactly what we see in our patients as well. So it gives us a good idea, perhaps, of what our patients might be sufferers of the Cornelia de Lange syndrome. So that's everything that I wanted to show you in the demo. Hopefully what you understand and what I've tried to demonstrate is how you can sort of use your own data that you've generated in the lab, or perhaps through a clinical report or something that you've been working on with some collaborators. You can upload your data to Ensembl, you can view it and begin visualizing it, and then look at it in the context of the other publicly available data that we have in Ensembl, like the gene annotation and the phenotype associations. And you can begin to connect the dots and to see how the data that you've had fits alongside all of the publicly available data that we know and we have in Ensembl. Good. Okay, so it's now 11:35. So what I want to do is give you 20 minutes to have a go at the exercises for this module. So if we go back to the training materials, if you didn't have a chance to have a go at uploading the bed file, if you weren't following along with me in that session just now, then please feel free to have a go at using this data and uploading it for yourself. But then also there's a section called Custom Data Exercises. So please try and have a go at using doing either the exercise here called adding wiggle files to Ensemble bacteria or adding BAM files to Ensemble fungi. So please do have a go at doing those exercises as well now. And you can do those obviously by going to the Ensemble bacteria or the Ensemble fungi pages, depending on which species, which exercise you want to have a go with. Good. So again, I'm just going to take a short break and I'll be back again with Alina in a few minutes to answer any questions that you have if you get stuck, or anything else about what we've been covering today. So we'll come back again and we'll start our final session at 12 o'clock and we'll cover, but before then, please have a go at those exercises and Alina and I will do our best to help you if you get stuck. Okay, so for anyone else that is also trying to re-go over the walkthrough, what we did is from these training materials, we took this bed file. This is the bed file that we are interested in for our demo. This is just some example data taken from 3 patients. So this is a bed file that describes 3 different deletions in 3 different patients. So the first row is describing the deletion in patient 1. So we've got the start coordinate here and the end coordinate here. And then the chromosome is the first column. So we've got chromosome number and then start coordinate and then the end coordinate. And this is the deletion for patient 1. And then we have the deletion for patient 2 on line 2. So we have again the chromosome number and then the genomic coordinate for the start position and then the genomic coordinate for the end position. And then on row 3, we have the deletion for patient 3. Again, the chromosome number the start coordinate of the deletion, and then the end coordinate of the deletion. So what we do is we copy these 3 lines of data, and then we open up Ensembl. So we go to www.ensemble.org, and then from here we need to search for our species of interest. So we know that the data that we have, the example data that we're using, is from a clinical setting, so it's human data. So we go to this link here on the left of the page, of the homepage, view full list of all species. And from here, you can begin typing your species of interest in the filter box in the top right-hand corner. So Homo, for example, for Homo sapiens, and then click on the link for human in the table. So this takes you to the species specific homepage. And then from here, sorry, are you still up with me there, or do you want me to go back a step?
I'm with you.
Cool. Okay. So from here, this is the species specific homepage for human, and there's a box in the top left-hand corner that's called Genome Assembly. So you should see this box, and within that box, there's a link that says display your data in Ensembl. So if you click there, it will open up a pop-up window in your browser where you can upload the data. So the first thing that you need to do is give your data a name. And then in the box, you paste your data. And then the final thing to do is to use the drop-down menu to select your file type. So we had a bed file, so this is what we need to select here. And then we click Add Data. So once we've clicked Add Data, we should get a message that says that our file has been uploaded successfully. And then the next section, it says, it's asking us if we want to navigate to the region of the genome that contains our data. So the 3 deletions that we obviously that we added in our bed file. So then you can click on this link, and it will take you to the region in the Ensemble Genome browser where our deletions are found. So we saw this page earlier in the workshop this morning. So we have obviously the chromosomal overview at the top. Then we have this overview image here. But then at the bottom, what you should see, at the moment it says loading component. So this page is just still loading. But when it finishes loading, what you should see is the normal view with all of the default tracks, but then with an additional track called Noblekinmat, which is our data track. So that's exactly what we see now. It's loaded. So we can see all of these other tracks, as we saw before with our genes of interest. And then now here you can see that we've got this Noblekinmat track that shows us our 3 deletions. So we've got P1, P2, and P3. So these are the regions, these blocks that are labeled P1, P2, and P3. They represent the regions of the genome that are deleted in our 3 different patients. So you can see here, for example, P1, you can see the start of the deletion here, and it goes all the way to the end of this view that you can see. And you can actually see that this deletion is overlapping, or all 3 of these deletions, in fact, they're all different sizes and they have different coordinates, but they actually all overlap this gene called NIPBL. So what we can actually begin to understand is, or begin to hypothesize, is whether the deletions that we have observed in the patients and the clinical symptoms that they're presenting, whether they actually whether the deletion of the NIPBL is responsible for those the observations that we have for those patients. So we can actually click on the NIPBL gene, click on the link for the gene within the pop-up window, so the stable ID ENSG 0000164,190. And then that takes us to the gene tab, and we can begin to explore a little bit more about the molecular function and the biological process that this gene is involved in. And we can also see, if we click on phenotypes, we can see if there are any known phenotypes that are associated with this NIPBL gene. And in this case, we can see that this gene is indeed already associated with a disease called the Cornelia de Lange syndrome. And if we click, I won't do this now, but if you click through onto these OrphanNet and MinMorbid pages, for example, and DDG2P, you'll be able to see information about the Cornelia de Lange syndrome. And you'll see actually, yes, in fact, this syndrome is associated with deletion or loss of the NIPBL gene function. So we can begin to understand perhaps the molecular mechanism of the observations of the phenotypes in our patients.
Hello, sir. Sir, thank you very much for the explicit explanation. I was able to do it to that point. Hello, can you hear me?
Sorry?
Yeah, I said, please, am I audible?
Yes, yeah, we can hear you, yeah.
All right. I said, thank you so much for the recap that you did again. I actually had to follow through, and I got to a point that displayed that page where, okay, you begin to see the different points at which those deletions, were indicated on that bed file, that interface specifically. But for me, I didn't, it's not properly indicated ahead, this exactly. I don't know if I'll be permitted to share my screen so that I can show you what I'm trying to say. It's not, I don't know. Okay, I think I got to this point, but then I think I'm kind of stuck in between here. So I don't know if you can please do a little recap and then.
Okay, so is this also loading for you, or is it already loaded at the bottom? Because at the moment my page is reloading.
Okay, let me minimize and check again. It's just a minute, please.
Okay.
Oh, mine is done loading.
So does it look like my screen, or is yours still loading?
No, it's fully loaded. Like it's the page displays all that it's supposed to be on the interface, I guess.
Yeah.
So okay, I think I can see what's on your screen. Just I don't know, it seems mine is a bit, or maybe I need some clarification on that, really.
Okay, it might be that you need to like resize your screen or change the dimensions of your window, because as you will see, well, I'll do this now. As you see, if you sort of like begin to move your screen, if you change it, then obviously it like reloads and it sort of all the images all reconfigure to fit the screen. So it depends if obviously it won't be what you see will not be exactly what I see, because I'm using a different size screen with different like window sizes and all those sorts of things. So it might not be exactly the same, but as long as you can see a track that has the name, I mean, I use Noblekinmat, but maybe you used a different name for your track.
Yeah, I used a different name too for mine, yeah.
But if you can see the name of your track with the data displayed there as these blocks, then that's what you need to get up to in this sort of.
Oh yeah, yes, I can see. Oh yes, I can see it now. Okay, so I can see my P1, P2, P3, the deletions, right?
Yeah.
Okay, so what did you say we have to do at this point, sir?
Well, so obviously it depends. This is just one example. So this is one data type using one example data set. So what you actually want to do in your work when you're using Ensemble in the future, it might depend upon what data you're looking at. But in this case, we obviously are looking at some deletions that we've identified in these 3 patients.
Okay.
So, and these blocks represent the regions of the genome that are deleted. So what we're trying to now see is whether these blocks, whether these deletions, whether they overlap with any other features in the genome. So whether they overlap with any perhaps genes or regulatory features or other things. So what we're going to do is look down this screen and look at the other tracks and see if there's any features that overlap with all of these 3 deletions. So if we scroll down, this is exactly what we're beginning to see now. So now I'm looking at the genes track here. You can see it's highlighted just where I'm hovering my mouse.
Okay,
so we can see that these three, so it's important to remember that when you're looking at the screen, everything is aligned along the genome. So where my mouse is over on the left here, you can see this is a region of the genome that's deleted by P2, but there's no gene here. Whereas over on the right-hand side, you can see that this is a region that's deleted by all P1, P2, and P3. And that's actually a region where this gene called NIPBL is found. So you can see actually what's happening in these three patients is that there's regions of NIPBL that are being deleted, this gene. Now, you might not have heard of NIPBL before. It's not like a, you might not know if it's associated with any diseases or phenotypes already. So what you can actually do from this interface is click on the gene name, or in fact, you can do this with anything here. So you can click on any of the features in this view and get more information. So this is, if you click on the gene, you can get more information about the gene. So we click on the gene and we can see, okay, so we've got, this is the name of the gene. This is the stable ID of the gene. This is the location. This is the transcript that I clicked on itself. We've got a link to RefSeq. So that's interesting. We can click on this to see if there's any information from NCBI. And then there's also information about the protein that this transcript encodes as well. So if you want more information, then what you can do is click on the link for the gene, the stable ID here at the top. And that takes you to the gene tab. So once we're on the gene tab, we can begin to see if there's anything that's already known about the NIPBL gene that might implicate it in this phenotype that we're observing in the patient. So we can look to see if there's any gene ontology information using the links in the menu on the left here. Or what I showed everyone earlier is if you click on phenotypes, you can see if there are any diseases or phenotypes that are already associated with the NIPBL gene. And indeed, when you look here, you can see that there are three databases, OrphanNet, MIM-Morbid, and DDG2P. These three databases all associate this NIPBL gene with a disease called the Cornelia de Lange syndrome. So if you might not have heard of this before, so if you're not sure, you can click on the links for the source data, which is just here in the table. So if I click on MIM-Morbid, for example, I can go out to the MIM-Morbid pages, and I'll see here more information about Cornelia de Lange syndrome, including a description of the presentation of this syndrome, and even any genetic heterogeneity and clinical features as well. So you can see Cornelia de Lange syndrome caused by a mutation in the NIPBL gene. So you can see actually this is already, this gene is already associated with the Cornelia de Lange syndrome. And in fact, so therefore, what we might be observing in our three patients is how is the deletion of this NIPBL gene that might be resulting in Cornelia de Lange syndrome in these three patients.
Thank you very much, sir. So my question is, now that we discovered that the deletion is what actually causes that particular pathological condition at its way.
Well, so I just wanted to correct you for just a small point there. So when Ensemble is not a diagnostic tool, so we haven't proved that this deletion means that these people have Cornelia de Lange syndrome. Like there's obviously, this is information that we could feed back to a clinician or to the physician, that the doctor that these people have, that these patients have. So then we might be able to actually do some diagnostic tests. But this is a way that we can begin to understand the genomics underlying perhaps what these patients are presenting with.
Oh, wow. Okay. All right. Thank you. I get it now. Thank you so much, sir.
Okay. Yeah, no problem at all. Yeah. So there's one question there's a question from someone called Sunday. Maybe, I don't know if you want to type your question, Sunday, or if you're happy to unmute yourself and you want to ask your question aloud.
Okay. I think, thank you very much. You know, I don't have a system, but I just have to learn. So I have an Android phone and I'm using the desktop site. So I think it took me much time to be able to zoom out. So I was thinking maybe it's an error from my end, but I think maybe it's because of the power of my phone. So
so
I'm trying to still learn. You know, I think one of the things I know that is very important in all of this analysis is actually is the interpretation. And I think that's what I'm trying to learn here. And I understand the fact that this is a database, can give you as many information that you can use to maybe help the doctors in diagnosis. You know, so I think I'm learning something, even though I don't have a system yet.
Okay. Yeah. So just a couple of things to mention then. So, so Ensemble is an online genome browser. So, if things are going slowly, it might be that there's problems with our server, or it might be that maybe your internet connection is maybe a bit flaky, or maybe even you're trying to load a page that's got a lot of data on it and it's just taking a few minutes. So you could even see for me, some of the pages were taking a minute or two to load, and that was because I'm obviously asking Ensemble to return quite a lot of data to me. So if things are taking a bit of time to load, sometimes the best way is just to be patient and to see if it corrects or loads in a minute or two. But sometimes it can be down to like your internet connection as well, because it's an online genome browser that works over the internet. And secondly, so you are correct. Like it's Ensemble obviously Ensemble is a tool and a database in the way that we present data. There's so many different types of data, and that we don't expect everyone to use every single type of data, but it's therefore down to the scientists and the people who are using Ensemble to navigate around the website, find the data that they need, and then to sort of begin to understand what that data means, and then you can export it and begin analyzing and processing it. So Ensemble is not the only piece of the puzzle when you're sort of doing your work. It's sort of maybe the starting point where you get some data, you can export it, and then you can begin making hypotheses and doing some analytical steps, and then designing your own experiments and designing your own follow-up experiments after that as well. So yeah, you're exactly right, Sunday.
All right, sir. I have a question, sir.
Yeah, sorry. Who is speaking? I can't see who's.
Effiong.
Okay. Hi,
all right. Okay. You know, you've displayed three samples, P1, P2, P3.
Yeah.
And these are deletions that you said were detected from 3 patients. Now, my question is, how did you come about this detection? Is it that you did multiple sequence alignment and you were able to detect the detection? And this detection, is it a kind of single nucleotide, or is it a whole deletion, or is it a gene deletion or nucleotide deletion? How did you come about this deletion, sir?
Well, okay, so the first thing to mention obviously is like the example that we used is made up. So it's I didn't actually get this data from anywhere. It's just it's made up as an example. But if you were to imagine how this would work in real life, it could be that what's probably happened is that these 3 patients who've presented with these, they've come to a clinic, maybe they've got these particular symptoms, and the physician doesn't exactly know what's wrong. So maybe basically some genome sequencing has been performed. So sequencing can occur at lots of different levels. So you can perform whole genome sequencing, you can perform whole exome sequencing, or you can even perform sort of targeted sequencing. But this obviously depends upon, firstly, like your budget, your time, your ability to analyze the data, and how much you already know about the patient. So for example, if you had these 3 patients, as the physician, if you had these 3 patients and you spoke to the families and you got some history of the patients and you knew that that a number of the members of the family already suffered from Cornelia de Lange syndrome, then you probably would already know or have a good idea that the area to begin looking in would be the NIPBL gene. So you could actually do some targeted sequencing of the NIPBL gene, perhaps, and begin to understand, like, without having to do whole genome or whole exome sequencing. Whereas obviously, if you have a patient where you don't understand the history, or you can't even begin to think about where to look in the genome, if you don't know which gene to begin doing some targeted sequencing against, you could do some more general sequencing where you look across the genome, the whole genome, for example. So this is sort of, it depends on the context and what's happening in that individual there.
So I have a question.
Yes.
Okay. The problem I have now is I have some raw sequences.
Okay.
Converting them to a bed file format or any format that is accepted on the Ensemble is a problem, because I've been trying to Google it, how to convert, and I'm not seeing any good response. How to convert to bed file or to any of the file. Are there other formats apart from bed file, and how do we convert it? I guess that's my question.
Okay, so so I think the best thing for me to do, if you write your question in the chat box and put it there, me or Alina or I can write an answer in the chat box for you, because I think it'll be too difficult to really show you on the screen. I think it'll be easier to type out an answer with some links.
Okay, I should type the question.
Okay, thank you, Eric.
Did I type the question?
Yeah, in the chat box, in the Zoom chat box will be okay.
All right, I'll do that.
Okay, thank you. Okay, so I think that there's a few other comments as well. So Patricia, Ensemble website requires patience and constant practice to learn. So before we move on to the final module, like I will say, Patricia, you're exactly correct. So what we've covered today is just a very, very brief overview of some of the things that you can do with Ensemble. So there's a lot more things to explore, and there's a lot more data to look at and to understand and to uncover. So what I'm going to do at the end of the session today is show you some further online courses that you can access and you can take to continue learning about Ensemble and to continue looking at other resources and learning and after today's session. So hopefully you have seen how useful it is and that you want to continue learning. Okay, so what I want to do now is go on to the final module for today. So the final module is a wrap-up where we just want to cover some of the things that we thought about, and then we'll also have some time for some more questions and answers as well. And I also want to show you some other resources that will be useful. So the first thing that I'd like to do is ask you to all fill in the feedback survey for the course today. So the first thing I'm going to do is go to the materials for the course. I'm going to paste this into the chat box. And from here, you can actually see that there's a link for the survey. So if you click on survey, this will take you to a page, which I can also give to us as a link in the chat box for today's workshop. So what we'd really appreciate is if you could spend a few minutes now to go through this survey and answer the feedback and provide some feedback on the workshop today. So please do fill in these. Please fill in this survey. It should just take a few minutes. At the bottom, if I get there, at the bottom, there is also an option for a certificate of attendance for this course. So if you click yes here, when you click next, you'll be prompted to enter your name and your email address. So you'll be able to receive an automated certificate of attendance for the course today.
I'm sorry, Doc. Please,
How do we get our way to get here?
So you can follow the link in the chat box. So if you go to the Zoom chat box, you can click on this link that I've provided in the chat box, and that's how to take you to the Google form. So you just need to.
I've sent the question to the chat box.
Pardon?
Sent my question to this chat box.
Yes, yeah. I'm going to get to that, and I'll answer it with Alina in the question answer session at the end of the session today. So what I'd like to do now is just give you a few minutes to fill in that feedback survey. We'll come back together in just 3 or 4 minutes time, and hopefully you'll have done that, and then we can continue with our wrap-up module.
Please, I have a question to ask, even though I'm already filling the survey.
Okay, so can everyone see my screen?
Yes, I can see.
Okay, so you need to go to find the training materials for today. The link is in the Zoom chat box. And then from the material, click on the link that says survey. And that will take you to the Google form. And I'll also put the link for the form in the chat box as well. Okay, so there's a few people that are already mentioning that they've completed the survey. So thank you very much for spending a few minutes to fill in the survey. What I'd like to do now is just move on to present some more slides and then also open the floor and answer any final questions. Oh. So just to wrap up what we've covered, so the main thing to remember is that Ensembl is a genome browser which integrates these 4 main types of data. So we've got the gene annotation, which we spoke about the first thing this morning. We also have the variation data, the regulation data, and the comparative genomics data. So we didn't look at these 3 other main data types in any real detail, but there is information there that there's some online courses that are available that will walk you through these other data types, and I can show you them as well at the end. So all this data is organized in a number of ways. So the main way to access all of this data is through the Ensembl Genome browser website. So we've got the main website, and then we also have the Ensemble Genomes website as well for the non-vertebrate species. If you want to access this data on a larger scale, then you can also consider using the Bioinformatics data mining tool. We have the Ensembl databases, which you can access using the Perl and the REST APIs. And you can also download genome-wide files using our FTP site as well by following the link here. So if you want further help and documentation, there's a number of ways that you can get that for using Ensembl. So the first is through our online courses. So we have a number of online courses that are all provided through the EBI training platform. So Ensembl is one project that's part of a larger organization called the EBI, the European Bioinformatics Institute. So I just want to spend a few minutes now just navigating to the EBI pages and showing you about the tools and resources and training that we have there. So if you go to www.ebi.ac.uk, I can put the link in the chat box again. This takes you to the EBI homepage. So from here, you can find out all of the information about the tools, the resources, and the training that we have available through the EBI. So the first thing that you can click on in the black bar at the top is the services link. So if you click on services, you can see information about all of the different tools and resources that we have in the EBI. So we have information about featured data resources. So we've got AlphaFold for protein structures. We've got biomodels. We've got Chembl. Then you can also see that we have featured tools. So we've got Clustal Omega for doing alignments, Hammer as well as a protein homology searching tool. But you can also begin searching here as well, or you can search, or you can explore all data tools and resources as well. So if you click here, you can explore all of the different resources that we have. So for example, we have Ensemble, which is a genome browser and gene annotation database. We have Europe PMC, which is a literature archive like PubMed. We have Expression Atlas for gene expression data. We have Intact, which is a curated database of molecular interactions. Reactome for biomolecular pathways in data. Uniprot for proteins. Complex Portal, Kebi. Basically, I won't go through all the different databases, but the idea is that we have different databases for different types of biological data. So Ensemble that we've been looking at today is for genes and genomes. But if you're interested in proteins or molecular interactions or chemical entities, there's lots of different databases that you can use and you can access through the EBI platform. The other thing that you can do through the EBI is to access the online training that I was talking about before. So if you go back up to the top of the page and click training, you can find all of the different online courses that are available through the EBI. So you can search, for example, if you just begin typing Ensemble and click search, you can find all of the different Ensemble courses that are available. So these are all online courses. They're free of charge. You don't have to pay to take them, but you don't get any accreditation or certification for completing the course. It's just an online course that helps walk you through some of the different data and resources that we have. So we've got the Ensemble courses. We've got a webinar series that you can watch the webinars. We've got an online course that walks you through Ensemble, and we also have some Ensemble Genomes workshops as well that you can watch about using the non-vertebrate Ensemble Genomes data. So let's say, for example, you wanted to take this course, Ensemble Browsing Genomes. All you'd do is click there, and you can see an option to enter the course. You can see that it's going to take you 3 hours approximately, and it includes activities, quizzes, and videos that you can watch. So then you just go to enter course, and then you can begin going through the course. And this is the book on the left-hand side that you go through the course book. So again, this just is these courses will hopefully build upon what we've covered today and help to cement the knowledge that you have from today's workshop, and then continue learning and using about some of the other data types that are available in Ensemble. Good. The other thing that you can do to continue learning about Ensemble is to visit our YouTube channel. So again, I'll just show you this in the browser quickly. If you go to YouTube and type Ensemble training, you can go to the Ensemble training pages and find all of our videos for that relate to Ensemble. So we have a number of different tutorials, recorded webinars, and recorded workshops that we have uploaded here that allow you to begin learning about Ensemble and the different data and tools that we have as well. So many of the things that are in these videos we would have covered in the course today, but there are other things as well that we haven't had a chance to look at that you can learn about through these videos as well. And the final thing that you can do is you can contact us by using our help desk and our mailing lists. So if you have a question about using Ensembl, you can email this address here. So it's helpdesk@ensemble.org. If you have any questions, you can email that to us, and we'll always get back to you trying to help you with any questions that you have about using Ensembl. If you can't find something, or if you're not sure what some of the data is, what it represents, then we can also talk you through that as well through the help desk. You can also subscribe to these messages, to these mailing lists here. So we have dev@ensemble.org and announce@ensemble.org. So these are mailing lists where you can learn about some of the announcements and updates from the Ensembl project. Some people already in the course today were asking about how you can cite Ensembl in your work. So if you do use Ensembl in your work and you want to cite the Ensembl pages in the database, then you can use our publication, our annual publication that we have in the NAR paper, Nucleic Acids Research. This year's one is called Ensembl 2022, and you can find the link and the DOI for it here. There's also papers for Ensemble Genomes if you're using the non-vertebrate sites as well. If you've enjoyed today's course and you want to arrange an Ensembl workshop at your institute for your colleagues and friends there, then you can contact us on the helpdesk@ensemble.org email address, and we'll work with you to try and arrange a virtual or an in-person training course in your country, in your institute. And if you want to keep up to date with Ensembl, then you can stay engaged with us through social media. So we have a Facebook page, we have a Twitter account, so @Ensembl you can follow, where we post tutorials, we post information about our new releases, we post any updates about maybe any downtime because of maintenance. These are all things that we put on Twitter. And then we also have a blog. So you can see on the right-hand side here, www.ensemble.info. This is our blog where we also have posts about all of the latest data that we're going to be releasing, all of our job adverts, any announcements about important updates and features that we're including in Ensembl. And then that brings me to the end of the workshop today. So this is the final slide with the acknowledgements. So these are all of the people that you'd have seen in the photo. So this is our last in-person retreat that we had before the pandemic. So it was obviously, you can see a large team of people that are all responsible for putting Ensembl together and generating and analyzing all of the data. So you can see this is the list of the people that you'd have seen in the photo. And then we also have our funders as well. So the Wellcome Trust, Ensembl, and a number of other bodies that fund specific projects as well. So without their funding, Ensembl wouldn't be possible. And it means that the data that we have and the resources that we provide are freely available for everyone around the world who wants to use them. So that's the end of the workshop today. I just need to take a quick shot of the screen. So there's a few questions that are coming in that's asking about the certificates that are being received. So the certificates are generated automatically. So they might just take a few minutes to come through and to be delivered to your email address. If after
mine has been delivered.
Okay, so I think the best thing to do is to be patient. So it might take even the rest of today. So if you still don't have a certificate by Monday, perhaps next week, then you can email helpdesk@ensemble.org and we'll try and arrange to get your certificate sent to you individually rather than automatically. So yeah, keep checking your inbox. It should arrive with you by the weekend. So if you don't have anything on Monday, then you can contact us and we'll arrange that individually for you. Okay, so what I'd like to do now is, it's now 12:30. So I just want to have the last maybe 10 or 15 minutes to answer any final questions. So the first question that I can see that's come in is from Daniel. So it says, in what ways can Ensemble assist a metabolomics researcher? This is quite tricky because, as I've mentioned, Ensemble is a database that's dedicated to genes and genomes. So I think metabolomics is a lot more related to sort of pathways and reactions and enzymatic function. So I think what you'd probably more likely be interested in looking at are some of the other databases available through NCBI, through EBI, sorry. So if we go to www.ebi.ac.uk and you go to services, you can see, for example, so here we have, for example, a database called Magnify, which is a platform for the analysis and archiving of metagenomic and metatranscriptome data. So this might be interesting for you if you're interested in metabolomics. But we also have Reactome. So you've got the Reactome tool, which is manually curated database of molecular pathways. But then you also have a tool called the Reactome Pathways Analysis, which is a combined tool to identify pathways and overexpression analysis tools. So basically, I think the thing to do is go to the EBI pages and see if there are any tools that will help you in your metabolomics research. The next question is coming in from Sunday. So it says, I want to ask if there are opportunities for internships for a postgraduate student interested in bioinformatics. So again, through the EBI, so directly through Ensemble, this is more difficult. It's normally arranged through the EBI, the larger organization. So if you come to the EBI pages, at the very bottom, there's a link for jobs. And here you can see information about all of the jobs, but then we also have links for internships and then PhD program and postdoc opportunities as well. So I'll put this link in the chat box and you can explore the different training and internship programs that we offer. And then the next question is coming in from okay, I think I'm going to mispronounce your name, but I'm going to try my best. So Abdurrahman. So the question is, I noticed Ensemble isn't yet a repository. Sorry, please correct my my butchered pronunciation.
Yes,
I didn't mean to get it wrong.
You got it right. You got it right.
Abdurrahman?
Yes, of course.
Okay, perfect. Yes. Sorry, like I do quite a lot of training with different people around the world, and I see so many different names from different cultures and countries that it's I don't always get the pronunciation 100% right. So I apologize if I've got your name wrong in the course today. I do try my best. Okay, so but your question is, you noticed Ensemble isn't yet a repository for animal genomes. Is there work in progress on that? How can someone with an interest in genomic or precision medicine in the veterinary field benefit from Ensemble? So there are animal genomes in Ensemble. If we go to ensemble.org, obviously it depends on the species that you're interested in, but if you're interested in vertebrate species, so maybe agricultural species or domestic veterinary, like animal species like cat and dog and those sorts of things, then you can click on this link here, view full list of all species, and you can begin searching. For example, cow. You can see we have in Ensemble sheep. We have a number of sheep. We have pig, but also a number of different pig breeds as well. We also have, if you're interested in domestic animals, then we have cats. You can see here at the top. We have dog and a number of dog breeds as well. I'm not sure exactly what you're interested in, perhaps, Abdurrahman, but
horses.
Pardon?
Horses.
So we do have horse in Ensemble, Equus Calibus here. So you can access the horse genome assembly and annotation here as well. So you just click on the horse and you can begin searching just as you've done before. So maybe even this UQCRQ gene that we searched before, you can find the horse UQCRQ gene and begin searching just like we did before. So yeah, it depends on the species that you're working with. Obviously, horse, we have the genome annotated and present in Ensemble, so you can search for it just as we've been looking at the human data earlier on today.
Thank you.
Perfect, no problem. Good. Okay, so there aren't any further questions in the chat box. So I think what we can do there is we can leave the course there for today. As I said, if you do have any further questions, then please do not hesitate in contacting us by using the help desk email address. So I'll just pop that in the chat box. And you can email any questions if they come up after the course and you want to ask them, you know, if they only come into your brain after we've finished the call today. There's one more question. Amanu has got their hand up. I don't know if you have a question, Amanu. Would you like to unmute yourself, Amanu?
Hello, I still have a question, Eric. I still have a question.
Okay, Eric, if you just put your hand up, because I think there are a few other people with their hands up, so then we can just make sure that we get to everyone. So,
hello, good afternoon, everyone.
Hi, is that Amanu?
Yes.
Hi, do you have a question?
Yeah, my question was about the exercise for today. I want to know if maybe at the end of the session today, and maybe because I'm not with my laptop now, so maybe when I do it, I can still submit and maybe a review will be done on it.
So we don't accept submissions of exercise answers, and we can't mark any of them. What you can do though, if you're working through the exercises, say for example you're having a go at the ones here, we do have sections with the answers and the answers. So we've got the text and the video answers. So you can actually review your answers yourself and see whether what you did corresponded with our model answers and how we got those as well. So this is a good way for you to check and to review your own work, I think.
Okay, thank you.
Yeah, no problem, Aminu. And then the next question comes from Zubeki.
Sorry about that.
Sorry.
Okay. Mistake.
Yeah. Do you have a question? Oh, mistake. Okay. The next question comes from Akin Meiji.
Okay, Doc. Well done, sir. Thanks for your presentation.
No problem. Yeah. Do you have a question?
I have a question, sir. My question is, how about applicable number, or is not applicable on this and
sorry, Akin Meiji, I don't mean to be rude. I really can't hear you very well. I think there's quite a bit of background noise and there's a bit of breaking. So is it possible to write your question in the chat box?
Sir.
Sorry, I really can't hear you very clearly. I think it just might be the internet connection, perhaps. Eric.
Okay.
Sorry, yeah. Do you have a question?
Yes, I do.
Perfect. Yes, please go ahead.
All right. Thank you so much for the presentation. It's eye-opening for me, and I've come to love the Ensemble platform. Well, I think the challenge I'm having now is, excuse me. I think the challenge I'm having now is the conversion of the files, because I have my own raw sequence data I would love to try out, apart from the exercises given on the workshop platform. But I also love try out. I've tried it out. I've tried out the exercises. They all worked perfectly. I really love the display. But I also love to try out with my own sequences, raw sequences I have, and they're in faster format. And I've been trying, though I saw your answer you gave, but it's not quite explanatory.
Okay.
But I don't know, is probably the Ensemble platform can accept faster format, because there's a place in the survey where they said if there are other challenges, and I wrote down that if it's possible to make the Ensemble platform accept faster file formats, or the conversion as well, to probably, I can get lessons on how to make that conversion. I really love to.
Okay, so I think the best starting place for you will be the BLAST tool. So I think so Ensemble doesn't accept FASTA because so FASTA is its own sequence, and what we have in Ensemble is also sequence. So all of when you're uploading the other file formats like we did in the example, that's all being aligned to the reference genome or mapped to the reference genome. But in the case of the FASTA, what you actually need to do is you need to find where that sequence maps to the reference genome, right?
Okay.
So what I would probably advise you to do is either use another alignment tool such as Omega. So if you go to EBI, you can also try using this Cluster Omega tool.
Okay.
So I'll put that into the chat box.
Okay.
But within Ensemble, you can also use BLAST. So BLAST stands for the Basic Local Alignment Search Tool. And if you click there, what you can do is you can input FASTA sequence here. For example, like this. I mean, this is just an example. And then you can use the BLAST tool to find where this sequence aligns to in the reference genome. So what you're saying is, like, I've got this sequence and I want to find where that aligns to in the whole of the human genome, whether that corresponds to a fragment of DNA or a gene or anything like that. So you can use the BLAST tool to search for your FASTA sequence in the reference genome sequence as well.
Okay,
so that's what I would begin to look into. I'd look into Cluster Omega and to the BLAST tool, and they might be able to help you take your FASTA sequences and begin entering into Ensemble that way.
Okay, thank you so much. I will try it out. I really hope I can get back to you. How do I get back to you if I have any questions or challenges?
So it's the best place is through the Ensemble help desk. So if you have questions, I'm just typing the address in the chat box now. If you write an email to helpdesk@ensemble.org, it will create a ticket in our system, and then we will be able to reply to you that way.
Okay, thank you so much. I really appreciate it. Thank you so much. I'm very grateful.
No problem. No problem. The last question that I have here is just, I think it just comes back here. I got tripped off at some point. What do I do to get the certificate? So to get the certificate, you just need to fill in the feedback survey, which is here. So here is the feedback survey in the chat box. So if you fill in the survey, and at the very bottom, what you will find is there is an option to select, would you like to receive a certificate? So if you click yes, you will then be asked to supply your name and email address, and you should receive your certificate via email automatically. As I said, I think some people it's a bit delayed. Sometimes it takes 5 or 10 minutes, or the survey link isn't working. Okay, it should be working. So I would advise you to maybe come back to the materials later on and then try and access them again. So if you go to the materials, which is linked in the chat box, you can access the survey through there as well. So if it's not working for you right now, then maybe you can come back to the survey later and see if it works then. Maybe it's just that it's inaccessible or for you at the moment. Okay, perfect. So that brings me to the end of the workshop today. Thank you so much for all attending. thank you. I hope you found the workshop useful, and I hope you're interested in using Ensembl more in your work in the future, and for learning about Ensembl using all of the other resources that I've showed you as well into the future. So we'll finish there for today. Sunday, the certificates might take a few moments, so if you haven't received one by next week, then please contact us on the help desk, the Ensembl help desk, as I mentioned. Yeah, so thank you very much for attending, and I hope you found it useful, and we will see you soon. Hello?
Thank you so much, Doctor, for the time, and Dr. Alina also. Really appreciate you taking out your time to take us through the Ensemble Genome browser. Thank you so much. This is the second time we'll be inviting you, and we hope that soon we'll have another session that we'll have to invite you again. So we're looking forward to that time. Thank you so much. Really appreciate your effort.
Thank you very much for hosting us, Ashraf, and we look forward to working with you again in the future.
It's our pleasure.
Thank you. Bye-bye. Bye-bye, everyone.
Bye-bye.
Bye, everyone.
Bye.